num quarto de hotel com o marzão do lado de fora.
Azul, mostrou pra gente, falei, nossa, que delícia, né?
Nossa, e eu aqui no meu quadrado.
E deve estar tomando breakfast dele agora, né?
Acho que sim, eu não sei se tem um delay, mas... E você, Marcelo, vocês enfrentaram o Covid? Foi tudo bem?
Foi tudo bem. A gente ficou... Foram no quarto dia de bastante febre, assim, sintomas de uma gripe bem forte.
Nossa, Marcelo, você teve? Eu não sabia.
Tive.
Nossa.
De uma gripe bem forte, assim, com febre alta, 38, 39. E que eu estava baixando, aí eu já fui no pronto-socorro aqui de Santana de Parnaíba mesmo. E aí já cheguei, ele já... é sintoma de Covid, já vem para cá, já fizeram toda a avaliação, né, do pulmonar principalmente, mas nenhum de nós estava com o pulmão comprometido.
Nós já estamos no YouTube, viu? Então todo mundo está sabendo que você tem.
E aí já começou o tratamento profilático com azitromicina, e que foi o que eu acho que segurou, assim. E aí os sintomas, o mal-estar, a febre, essas coisas todas passaram rapidamente. Assim, 2, 3 dias depois já tinham passado. Mas o que ficou foi uma fadiga, assim, sabe? De vez em quando dava um cansaço, uma moleza em que você era obrigado a largar tudo, deitar e dormir, assim, não importa a hora, 2 da tarde, 3, acabava a energia de repente, assim. E isso durou 10 dias.
Que coisa, né? Bastante.
E a Rita teve bastante problema de olfato, ela perdeu o olfato, e o dela tá voltando agora, assim, 1 mês e tanto depois, só que tá voltando.
E, Marcelo, você sabe como pegou? Foi por conta de máscara? Eu queria entender como é que estão as pessoas pegando agora? Qual que é o...
Não temos a mínima ideia, porque a gente só sai de casa uma vez por mês para fazer compra, no mesmo supermercado, máscara com álcool em gel, como a gente sempre fez. Pegou.
Que coisa. É isso que eu fico... Eu achava assim, bom, às vezes encontrou alguém, né? Teria que ter sempre...
Então, mas
que você saiba, né?
Na Globo News que essa segunda onda que está acontecendo, ela está com essa característica, assim, as pessoas não estão sabendo onde estão pegando.
Que coisa. Que coisa. Ainda bem que você me avisou isso, bom saber, viu?
Não tem uma coisa, não tem um marco evidente, assim, não, olha, fui numa festa e peguei.
Que coisa.
Tem muita gente que está em casa, não está saindo e pega.
Bom dia, Cláudio.
Olá, Cláudio, bom dia.
Bom dia, Irma, bom dia a todos.
Bom dia, Sylvio, como vai você?
Bom dia.
Mais uma vez, um prazer revê-las.
Só avisando que a gente já está ao vivo, tá? Só para avisá-los todos que estamos já no YouTube.
Essa nossa conversa inicial está passando.
Olá, Sylvia.
Sylvia.
Tudo bom?
Tudo bom.
Cláudio, tudo bom, Cláudio?
Tudo bem, Sylvio, e você? Como é que está?
Na luta, né, Cláudio? Tentando ver se tem luz no fim do túnel mesmo. Mas, o Ema, eu estava pensando nesse tema aí, esse negócio de gêmeos é interessante, viu?
É muito, Sylvio, muito.
Muito. São os iguais que são diferentes, né?
Isso. Eles são interessantes pelo que são parecidos e são interessantes pelo que são diferentes em tantas coisas, né? Em saúde, em aspectos comportamentais, psicológicos, eu acho que é um mundo, né?
É um mundo. É um mundo.
Sim, é um tema fascinante, sim, esse tema é maravilhoso. quando a gente começa a se envolver mais, né? A quantidade de perguntas muito interessantes que vão surgindo, dá vontade de ter 18 braços, ter 5 pessoas na equipe e abraçar todas as perguntas que vão aparecendo.
O Cláudio está bastante engajado na análise dos dados de voz, sabe, Sylvio? Então, a gente tem gravações da voz de cada um deles, né? E então nós usamos um programa que chama Prate, que vai fazer essa análise de parâmetros acústicos, né? E depois tem um pessoal lá da computação de São Carlos, que também está fazendo uma análise com um olhar um pouquinho diferente, usando inteligência artificial, né, para tentar ver a distância entre esses parâmetros da voz dos gêmeos monozigóticos e dizigóticos, né?
Esse é um assunto... pessoal do André, não? André Ponce de Leon? Lá do ICMC de São Carlos, né?
É o ICMC de São Carlos, mas não é esse professor, é uma professora, Sandra, né, Cláudio?
Sei.
Bom dia, professora Ana Loffredo. Estamos aqui todos reunidos e nós já estamos no ar, viu? Então, 8h59 aqui no meu reloginho.
Professora Ana, bom dia.
Eu estou falando à toa, gente. Bom dia para todo mundo. Eu falei bom dia para todo mundo e ninguém ouviu. Bom, nossa, EMA, 8h59, o evento organizado pela EMA, né, Marcelo?
É sharp.
Quando organizar eventos de psicanálise, eu vou chamar a EMA, né, Marcelo?
Mas parece que há grandes indicadores de que a pontualidade aumentou muito com esses eventos online, né?
Eu acho que sim, porque nós temos uma tradição, pelo menos no Instituto de Psicologia, não posso dizer, mas a gente atrasa muito, né, Marcelo, nos nossos colegiados e tudo.
É uma tradição ruim, né?
Uma tradição...
Não é o desejado. Mas, enfim, de fato, a gente tem mesmo esse péssimo hábito.
Mas eu acho que está melhor, viu, professor Canuto? Eu concordo, acho que a gente...
a gente tem que aprender...
Eu acho que tem. Pelo menos a minha experiência... Eu tenho feito muitas aberturas, como vocês podem imaginar. E a gente entra 5, 10 minutos antes, as pessoas já estão lá na sala, trabalhando, tudo.
É isso mesmo.
O professor Cláudio vai fazer a abertura, quer dizer, vai chamar os nossos convidados.
Yes.
E a gente faz em inglês ou português, Ana? Ou Emma?
Agora seria em inglês, porque nós temos os convidados internacionais, né?
Foi o que eu imaginei.
Well, good morning to all of you. It's a real pleasure to have all of you with us. We are opening the Twin Studies in Behavioral and Health Research, current status, perspective, and applications, a scientific event that we are making today here at the University of São Paulo. But we have people participating from all over the world. So I would like to say good afternoon and good evening to all of you who are with us today. This event is a part of 2 events. One today is a scientific event on research about twins. And tomorrow we have a more social event with twins, Brazilian twins, that's called Quinto, the 5th twins meeting of University of São Paulo. It's a long study, project that has many years so far, and it's a very important project that we have at the University of São Paulo. I would like to thank the presence of our Pro-Host of Research, Professor Sylvio Roberto Accioly Canuto, the Professor Ana Maria Loffredo, who is the Dean of the Psychology Institute of our university, and Professor Emma Otta, who is the organizer of this event. And now our staff, our team is here with us. So, I would like to ask Professor Sylvio to open this event with some words, welcome words.
Thank you, Professor Claudio Possoni. Good morning to everyone. Very nice to see you again. I'd like to thank the organizers for inviting me for this opening. Very nice, thank you. So I'd like to first salute Professor Ana Maria Loffredo, the Director of the Institute of Psychology, the Vice Director, Professor Gustavo Massola, and also Professor Marcelo Costa, is the head of the Department of Experimental Psychology, and of course, my dear Professor Emma Otta, and again, Professor Claudio Possoni. So I extend this compliment to the entire organizing committee for putting this meeting together. Very good meeting, great challenge. So I have to congratulate all the organizing committee for this. So on behalf of the University of São Paulo, I welcome all the participants, plenary speakers, and participants in general. Twin studies. This is a fantastic research topic. It certainly sparks our curiosity. So alike and sometimes so different twins are, I suppose. I remember a quote that said, "I may be a twin, but I am one of a kind." I think this is a nice quote. I wonder the diversity surprisingly involved in twin studies. I admit just beforehand, this is a fantastic topic. Congratulations to you all for organizing this meeting, and I am certain this will be a great success. I hope we will have a chance, not in a too distant future, to see you all in 3 dimensions, rather than 2 dimensions which are just faced on a computer screen. We're going to meet soon, I hope. So for this moment, this is the best we can do, and we cannot deny the facility to extend the geographic reach. These pandemic times will be remembered in the future. But they will know that the specific community, the scientific community, continue to work and to share knowledge using online system. And of course, this meeting is just one very good example. I am very happy and proud that our research office was able to partially support this meeting, and I wish you all a very, very interesting event. And before I conclude, congratulations again for all of you that work in such a fantastic topic. So be safe, and thank you.
Thank you, Professor Sylvio. And it is always time to remember and repeat that Professor Sylvio is always supporting us. We are very thankful to you and all your staff, your team are always very helpful, and you are supporting us in many events. Thank you very much, Professor Sylvio.
You are welcome.
Professor Ana Maria Loffredo, I would like to thank you to being here with us. We have a very large research group from professors, researchers from different units of University of São Paulo, and even other universities, just like Sylvia Porte, who's from Uruguay. But our group is hosted in your institution, the Psychology Institute. So thank you very much for being so kind host. And I would like you to invite you to say some welcome words for this event.
Thank you, Professor Claudio Possoni. Good morning. Welcome, everybody. I'm very happy and proud that our institute, with the support of the Pró-Reitor of Research, Professor Sylvio Canuto, is organizing with my dear colleague, Emma Otto, this so important scientific event with fundamental thematics from to the health area and to the developmental psychology area. Twin studies in behavioral and health research, current status, prospects and applications. Em português, estudos de gêmeos em pesquisa comportamental e de saúde. Situação atual, perspectivas e aplicações. I would like to express my gratitude to Professor Emma Otta, to Professor Claudio Rossi, and to all the team, all the persons who composed this team who have organized this event. And to the presence of Professor Marcelo Costa, Head of Department of Experimental Psychology, and to the presence of all speakers and participants from Brazil and from other countries. It's very important that in this so complicated moment we are living, research and scientific events are occurring, even in a virtual meeting. I would also like to express my gratitude to all the works of the Institute of Psychology for the technology support to the event. I wish all a successful meeting.
Thank you, Professor Ana Maria Loffredo. I would like to ask Professor Emma Otta, who is the creator of the USP Twin Panels, USP Twin Panels, to say some welcome words to her, to all of us. Professor Emma, please.
Claudio, I think it's Marcelo. No, it's okay. Sorry.
Okay. I would like to thank Marcelo Fernandes Costa for being here with us. He's the head of the department of where we are hosted. And Professor Marcelo, if you have some words to welcome us, please.
Thank you, Claudio Possani, Ana Maria Loffredo, Sylvio Roberto Canuto, and Emma Otta to invite me to this opening. I would like to give my good morning for all my dear colleagues. It's a reason of great pride for the Department of Experimental Psychology to host this international event, Twins Studies in Behavioral and Health Research, Current Status, Prospects, and Application. This is an innovative, fascinating research team that makes our department very proud to participate on behalf of Professor Emma and her research team. Also, this is a great opportunity to show the important contribution that experimental psychology has to the knowledge of the psyche and the human behaviors. Without much delay and hoping that we will take advantage of this opportunity for scientific dissemination, I wish you all a very excellent event. Thank you so much.
Thank you, Professor Marcelo. It's a real pleasure to have you here with us. Professor Emma Otta, please.
Good morning. My name is Emma Otta, and along with Dr. Tania Lucci and Dr. Claudio Postani, I represent the organizing committee of the event Twin Studies in Behavioral and Health Research, Current Status, Prospects, and Applications. I am happy to join with you today. We are grateful to Professor Sylvio Canuto, Pró-Reitor of Research of the University of São Paulo, for the support to the event. In the context of COVID-19 pandemic that has turned our world upside down, we stress the importance of creating and preserving opportunities for the communication and the exchange of ideas and research experiences among researchers. And we would like to emphasize the importance of the initiative of our university to foster new ways to hold scientific meetings. I would like also to express my gratitude to the presence of Professor Ana Maria Loffredo, Director of the Institute of Psychology, of Professor Marcelo Costa, Head of the Department of Experimental Psychology, and to the presence of all speakers and participants in the event. We organized it in the form of an interdisciplinary approach around themes of basic psychological processes and behavior, individual differences, well-being, and quality of life. I hope that it provides you with a forum to exchange scientific ideas, inspire new research, and new contacts for cooperation. I welcome the many participants from Brazil and from other countries. Some are not here today with us, but they have contributed in one way or another to our meeting. Professor Regina Brito actively participated in the years prior to the final creation of the painel USP Gêmeos that were important for the organization of our research network. Regina passed away in July 2020. On behalf of the organizing committee, I remember also Edila Aparecida da Silva, a laboratory assistant of the Department of Experimental Psychology at USP, one of the thousands of people who have died at age 62 with coronavirus in June 2020. COVID-19 pandemic has turned the world upside down. Vaccines are in development thanks to the efforts of scientists around the world. Science gives us hope for the future in our turbulent world. We are facing enormous challenges, but enormous crises also offer us opportunities. The pandemic forced us to adopt a virtual format, but it also lifted some barriers to participation in scientific events. I would like to express my gratitude to Professor João Eduardo Ferreira, the head of the University of São Paulo's Technology Information Superintendency and his team, and to Kátia Pinto and her team of the Information Technology section of the Institute of Psychology. I wish you a successful meeting. Thank you.
Thank you, Professor Emma. Well, so I am closing this opening session, and the next session is called Laying the Foundations for a Research Program. We'll have 3 talks, and a member of our team, Tania Chiaulusti, who is a very, very active person, who is one of the responsible for this event. We all are, thank you very much, Tania, for everything you did in these last weeks. Without your hard work, this event would not be possible. And Tania will be the moderator of this next session. So I ask you, Tania, to assume, and thank you very much for everything you did in the last weeks.
Thank you so much, Claudio. I'm very honored to have been invited to be a moderator of this session today, called Laying the Foundation for a Research Program. We will have, as Claudio told us, we will have 3 presentations in this session. Each presentation will have 15 minutes, and at the end, we will have 15 additional minutes for questions and general discussions around the 3 talks. The first talk will be from the organizer of the event, Professor Emma Otta, and Eloisa Fernandes from the Institute of Psychology of the University of São Paulo. And they are going to talk about the USP Twin Panel. Emma Otta is a professor at the Institute of Psychology of the University of São Paulo, and she is the coordinator and creator of the USP Twin Panel, Painel USP de Gêmeos. She had the dream of studying twins at the time of college and succeeded in putting it into practice. Today, the USP Twin Panel is in the 5th edition of the Twins meeting, and we have more than 5,000 registered. The studies associated with the USP Twin Panel aims to study the relationship between twins, attachment, cooperation, well-being through self-reported questionnaires, and also the study of emotions through behaviors observations such as facial expression, eye tracking, and voice analysis. The theoretical perspective adopted is human ethology. Eloisa Souza Fernandes is a psychologist that has collaborated with the group since the beginning of her career as a student. She's responsible for organizing the twin registration and has helped in numerous activities, being undoubtedly a key piece for the group. She's currently doing a master's degree with Professor Emma Otta on well-being in twins. I welcome both, and I will ask you to present your talk. Thank you.
Good morning. I will talk about the University of São Paulo Twin Panel. On behalf of the organizing committee, I would like to homage Regina. This talk is dedicated to Professor Regina Célia Gomes de Souza of the Center of Behavioral Theory and Research at the Federal University of Pará, who was a passionate and dedicated research mentor. She passed away on July 2020. Some dissertations supervised by her were adult attachment, life history strategy, and affective bonding twins, sexual orientations of twins in the north of Brazil. On behalf of the organizing committee, I would like also to homage Edila Aparecida da Silva. She was a laboratory assistant of the Department of Experimental Psychology at USP that hosts our event. Edila was one of the thousands of people who died at age 62 with coronavirus on June 2020. Vaccines are in development thanks to the efforts of scientists around the world. We hope that Butantan Institute in São Paulo and Fiocruz in Rio de Janeiro may help scale up manufacturing quickly for use in the country, these vaccines. Science gives us hope for the future in our turbulent world. Twins provide us with a natural experimental design to conduct research from behavioral and health perspectives. Currently, they are helping scientists to estimate the irritability of symptoms and to better understand why COVID-19 makes some people sicker than others. They are also helping psychologists to understand the factors involved in subjective well-being. Psychological responses to the COVID-19 crisis in twins are being studied by a team led by Robert who is comparing psychological and behavioral threats assessed 1 month after lockdown started and in 2018 before COVID. Preliminary results indicate that COVID-19 crisis that turned people's lives upside down did not change the individual psychologically in a fundamental way, but genetic differences played a role in shaping psychological and behavioral responses to the COVID-19 crisis. There are meta-analyses of the irritability of human traits based on twin studies. The most important is this one of 2015 based on 50 years of twin studies. It is based on 2,738 publications. I would like to stress that only 5 of these were headed by Brazilian researchers, and they are from the areas of genetics, medicine, and physical education. In spite of the importance of psychological studies in this meta-analysis, no psychology studies of Brazil is included in the meta-analysis. This is, in our opinion, a notable gap. I would like to stress that the knowledge produced as a result of the research being carried out on a global scale has an overrepresentation of some twin populations and an underrepresentation of others. Irritability is a property of a population. It is a statistical measure defined in relation to a particular population and a particular environment. Changing the population or the environment, the irritability may change. Brazil is the 5th most populous country of the world, economically heterogeneous, and a melting pot of ethnic and cultural diversity, with much to offer for research on twin issues. With the aim of fostering research on twin-related issues from a psychological perspective in Brazil and South America, we created the Painel USP de Gêmeos. It brings together twins interested in participating in research on basic psychological processes and behavior. We use social media to stimulate communication with and among twins. Twins or twin parents interested in participating of the Painel USP de Gêmeos can fill out a mobile-friendly registration form, which takes only a few minutes. We organize annual Twins Festival, and we are now at the 5th Twins Festival. We offer cultural and recreational activities for twins and spread evidence-based knowledge. Conversation circles have been attracting a lot of interest over the years. The main conversation topic among twins gives us a wealth of psychological insight. At our 5th festival on December 12th, Victor and Gabriel will be with us. They will sing and play guitar. Separated when babies, they met 24 years later. They are so alike as 2 peas in a pod. They met because one of them published a video singing on YouTube and the other watched. Both were gospel singers before they met. Their Facebook is titled Metade de Dois, Half of Two. At our 5th festival, Tomás and Gabriel also will be with us. Separated as newborns, they met 23 years later. One lives in Fortaleza and the other lives in Uberaba. They are also so alike as 2 peas in a pod. They were surprised to realize that both are photographers. One works at a museum and the other is a freelance photographer. The Painel USP de Gêmeos, formally created in 2017, has more than 6,000 individuals currently registered. 75% were from the Southeast, 15% from the South, 4% from the Northeast, 4% of the West, and 2% from the North. I will bring you a brief overview of research projects of Painel USP de Gêmeos. We started with population studies on twinning rates, and then began the validation of questionnaire-based zygosity assessment. We then conducted a self-report study on attachment. nonverbal behavior of twins are being studied, focusing on voice and facial expressions. I will also tell you how we are adapting our research protocols to the new remote work reality. So let's begin with our population studies on twinning rate in the city of São Paulo. We focused the period of 2003 to 2014 and found rates of 10 per thousand to 13 per thousand maternities. Then we examined twinning rates in Brazil in the same time period. The higher twinning rates were found in the southeast, 10 per thousand, and in the south, also 10 per thousand. And the lowest rate was found in the north, 7 per thousand, with the central west and the northeast in an intermediate position. Then we conducted a study to validate a questionnaire for zygosity assessment. The first step was to translate from English into Portuguese the 4-item questionnaire used by the Danish Twin Registry. We compared the results with the DNA in a Brazilian twin sample. The questionnaire correctly established zygosity with 96% of accuracy. This accuracy rate is virtually the same as reported by the Danish Twin Registry. In comparison, the accuracy of the self-report of zygosity was 60%. Here you have the questionnaire of the Danish Twin Registry and the Brazilian Portuguese version. Our next study was a self-report study on attachment. Our aims were to translate into Brazilian Portuguese the attachment features and function measures developed by Tancredi and Freire, to compare the relative rank of co-twins and parents using this questionnaire, and to test the hypothesis that monozygotics are more likely than dizygotic twins to use one another as attachment figures. This paper will be published in February of the next year, and it is already in press. Examples of the attachment features and functions, 16 items questionnaire, are these 3 items. My life would be severely disrupted if mine was no longer a part of it. It is important to me to see or talk with regularly. When I'm away from mine, I feel down. And the answers were given using a Likert scale of 7 points from strongly disagree to strongly. We are studying acoustic similarities and differences in the voices of same-sex, monozygotic and dizygotic twins. Acoustic analyses are carried out on vocal samples of 86 pairs of twins using the Pratt program. Each individual had his voice recorded at the lab in 3 conditions: saying, "Hi, my name is," saying the lyrics of "Happy Birthday to You," and singing "Happy Birthday to You." Examples of voice recorded analyzed by Pratt can be seen here. We will analyze the following measures of the speech of each individual: values of the first 4 formants, F1 to F4 in hertz, average value of the dominant frequency, the pitch, jitter and shimmer values, and duration. We are studying also similarities and differences in the facial expressions of same-sex, monozygotic, and dizygotic twins. Our twins were filmed while they watched happiness, disgust, and sadness-inducing films without seeing each other's reactions. Here you have an example of a film, a short video clip that we considered hilarious. and our twins also consider this. It is a baby that meets for the first time his uncle, that is a monozygotic twin with his father. He gets confused and passes from one lap to the other. Two dyads of monozygotic twins are watching this happiness-inducing video clip. There are individual differences that generate a facial expression signature for each person. What we want to know if there is a unique twin facial expression signature. One of the twin pairs is very expressive and the other is less expressive. The data are being analyzed with face reader. Due to COVID-19 pandemics, we adapted our research and shifted to remote data collection. We had started a study with children before COVID-19 pandemics, and we had already collected data with 10 pairs of twins in our lab. 5 to 8 years old children had been filmed during a joint task. They completed a puzzle. We replaced the puzzle by a drawing made together by the twins seated at a table next to one another at their house. The mothers completed an online questionnaire on twin relationship and then were invited to a second part of the research. And if they agreed, they received detailed instructions about how to film their children during a conjoined drawing task. We are looking forward to meeting everyone in person at the lab and in twins festivals, such as during this conversation circle that happened in 2017. However, we are grateful for the privilege of being part of the Painel USP de Gêmeos and for hosting this online event. Special thanks are due to Professor Regina Macedo, who painted this picture for us. And on behalf of the organizing committee, I invite you to attend the next talks of the event. Thank you.
Thank you, Emma, for your wonderful presentation. And now I will welcome Professor Sylvia Corti. Sylvia Corti is a professor at Faculdade de Ciências Universidade de la República, UDELER, in the area of ethology with specialization in primate behavior and ethology applied to animal welfare. She is the founder of the Twin Register in Uruguay and a close collaborator of the Twin USP Panel. She is coordinating a research on attachment and is currently preparing a study on the mother-infant relationship in twins. Today, she will talk about the Uruguayan Twins Project. I welcome you. Thank you, Sylvia.
Hello, everybody. Thank you, Tanya. Thank you very much. Today, we are here to share with you a project, the Uruguayan Twin Project. Almost the first studies on twinning in Uruguay. In 2017, we got in touch with the University of Sao Paulo Twins Panel due to the interest of a student, Noelia Gomez, in doing her graduation thesis on some aspects of human behavior. We couldn't find many studies of twins in our country, but a book that focuses on twins' generalities and a psychoanalytic treatment of twins and triplets, written by Dr. Miguel Cerro. Miguel Cerro is a pediatrician and child and adolescent psychiatrist, now retired. He also specialized in attachment. In July 2019, he was appointed an honorary member of the National Academy of Medicine from Uruguay. We invited him to participate today, but unfortunately, this time he couldn't. We had no other information regarding twining in our country. So Professor Emma proposed us to carry out a study on twining and thus contribute to the knowledge of the subject in Latin America. We managed to find some accessible data from government institutions such as the Ministry of Public Health of Uruguay. We focused on describing and analyzing twining rates in the country during the period of 1999-2015. Twining is not common among humans, although there is a great variability between populations. Some demographic and socio-economic factors influence the twining rate. Studies carried out by the Twin Panel in Sao Paulo showed that the twin birth rate increased from 10.19 to 13.33 per thousand births from 2003 to 2014. Twin rates have changed around the world, increasing since 1980s, probably due to the delay of pregnancy until the age of 30 or 40 years for mothers, and the use of assisted reproductive techniques. Furthermore, twining rates research in European countries determine that the most predictive factor for the multiple rate is maternal age. The present work, conducted from a human evolutionary perspective, intends to analyze different factors that could determine the twining rate in Uruguay and to study its variation between 1999 and 2015. If Uruguay exhibits the same behavior as other countries reviewed in the literature, it would contribute to finding reliable predictors. Where to get the data from? How to analyze them? We use data from the National Statistical Institute website, from censuses carried out in 1996 and 2011, and from the Ministry of Health. The birth rate was calculated for every 1,000 inhabitants in the region. The available data did not allow to differentiate between monozygotic or dizygotic twins. Monthly household income was defined by department. That is the way that Uruguay is politically divided. So the average for the region was calculated. The monthly per capita income was established for the urban area of each of the departments. Our software was used. The twin rate was calculated from the number of twin births according to the total deliveries in a ratio per 1,000, and the triplet rate was calculated in a ratio per 10,000. There is no data for triplets for the first 3 years. Between 1999 to 2015, the twin rate ranged from 8.51 to 13 for every 1,000 inhabitants. You can see also in this graphic the dramatic decrease of the rate of singletons that is characteristic of our country. We are a country of only 3 million people, and this is one of our problems. The rate of births in our country. The box plot shows the evolution of twin rates through time for each department in which Uruguay is politically divided. Montevideo, the capital city, has the highest median in the smallest variability in comparison with other Uruguayan departments. Durazno and Flores, at the center of the country, have the lowest median and the highest rate dispersion, respectively. It is worth noting that 43.7% of all twins were born in Montevideo. The region with the highest twinning rate is the metropolitan region, followed by the East region. In the 2011 census, the metropolitan region had the highest average household income. The twinning rate are more or less constant in more departments, depending on their population, but a marked increase is observed in the metropolitan region and some departments of the East and North region. East and North region. In all regions, the highest number of births in general occurs to mothers with complete primary school and incomplete basic cycle. As the educational level increases, the number of births decreases. In this study, more cases of twins were observed in mothers with a high level of education, more than expected, at least 12 years of formal education. In this figure, we see the highest twinning rate, 28.94%, that was observed in women aged 45 years or more. This could be due to the low number of total births registered for women of that age, that was 0.17%. Another possible explanation is the use of assisted reproduction techniques, ART. Older women may be those who have turned to ART as a last resource to have children. These techniques could be the cause of unnatural multiple birth. Also, older mothers would have better educational levels and more income, favoring access to assisted reproductive techniques. In Uruguay, and in the studied period, the average rate for twins and triplets was 10.9 per 1,000 births and 1.5 per 10,000 births, respectively. There was an increase of twinning rates, as was registered for other countries. The age of mother is a relevant variable, and as well as certain factors related to economic condition of the mother. In conclusion, this study recognized social, economic, and demographic factors that influence the rate of twin births in Uruguay. How many monozygotic and dizygotic are they? The National Registry do not specify zygosity. The birth is registered individually and catalogued as multiple. So our goal is to carry on an Uruguayan twin register that includes a zygosity questionnaire already validated by the University of Sao Paulo Twin Panel, the zygosity questionnaire of Christensen et al. 2003. Here we can see the Spanish version from the original English version. And you can see here that the original version was used in the Danish Twin Registry. And the first question is, do you and your twin look like two peas in a pod? And we translate those to two water drops, because it's the way it's better understand in our country and in Spanish when we wanted to know if you are very similar, almost indistinguishable. So in the Spanish version, we change that question for tú y tu hermano gemelo son como dos gotas de agua. For the collaboration with the USP panel, well, we intended to create an Uruguayan Twins registry to facilitate recruitment for research. The methodology would be online through social networks and request disclosure among acquaintances. We will follow the work done by Eloisa de Souza Fernandes para Panel de Gêmeos. El Panel de Gêmeos also have a registry, and it has been translated into Spanish by us. It includes the zygosity questionnaire. The question on socio-demographic issues are being revised to adapt them to the Uruguayan reality. For that, we ask collaboration to Professor Matthias Nathan of the Faculty of Sociology. We have another ongoing research proposals, some about attachment between adult twins, monozygotic and dizygotic, kin selection, satisfaction with life and perception of attachment with his twin brother or sister, always using questionnaires proposed by the twin panel, being translated from Tancredi and Fraley, 2006. These research proposals are being by Valentina Melo Gonzalez, an advanced undergraduate student of human biology, in collaboration with Dr. Laura Stern, a psychologist from the University of the República of Uruguay. Both the project and the informed consent form were approved by the Ethics Committee of the Institute for Biological Research, Clemente Estable, from the Ministry of Culture of Uruguay. 135 twins answered the questionnaire, a good number for a country with 3 million people. Later, we will have more info about this work in progress directly from Laura and Valentina. Further collaboration. We are collaborating in the design of an investigation of cooperation and competition in twins. This research is from studying drawings made by children between 5 and 7 years old, and is done using citizen science. Citizen science is the practice of public participation and collaboration in scientific research to increase scientific knowledge. Through citizen science, people share and contribute to data monitoring and collection programs. Citizen science initiatives have consolidated themselves as a new form of interaction between professional scientists, allowing them to participate in activities of formal research, providing opportunities for greater engagement in science. We are now in stage of translation into Spanish of the video and the questionnaire used for applying this research in Uruguay. For the panel of Gêmeos, the study is carried on by Luisa García Maschu and Carolina Greco de Almeida. A multidisciplinary approach. The Uruguayan group of studies in twin is looking forward its confirmation with a multidisciplinary approach. Biology, ethology, sociology, psychology, statistics, anthropology are the disciplines that we try to amalgamate in this process. We are interested in making an ethological approach to twins, especially on babies and children, based on direct observations, following the leader studies of dear Dr. Nancy Siegel. We thank the University of São Paulo Twins Panels for their support and constant motivation for us to achieve our goals. Thank you.
Thank you, Sylvia Corti, for your wonderful presentation. And now I would like to welcome Professor Ariel Nafonoa. Ariel Nafonoa is Professor of Developmental Psychology at the Hebrew University of Jerusalem. He is part of the longitudinal Israeli study of twins list that completed 17 years and which has around 1,000 families of twins from the age 3 to 17 years old. He studies the genetic and environmental influences on prosocial behaviors using the twins model and has demonstrated in his work how genetics and the environment work together in forming an empathic and prosocial disposition. I welcome Professor Ariel Nafo, and I would like you to present. Thank you.
Good morning. I'm very happy to be here. Thank you, the organizers, for inviting me. And this work is in collaboration with Dr. Dana Würzberger, who's now at Stanford University. And would like to present some information about our longitudinal Israeli study of twins, where we've studied the moral development of children in what is possibly the most comprehensive study of children's moral development using behavioral genetic techniques. So longitudinal Israeli study of twins includes over 1,000 pairs using a multivariate set of data with different ends depending on the different tasks that we had at every single age. And we're now starting our age 15 data collection. And when I say moral development, it could mean many different things. A lot of research is focused on age differences in moral development as compared to individual differences. Some other research has looked at moral reasoning, the more cognitive part, as compared to pro-social, the more behavioral part, moral emotions as well. Values, this has been hardly studied with children, but with adults, we have lots of work on values. So it's a very, very broad construct. And indeed, one way to look at it is to talk about different emotion, cognition, and behavior aspects of morality, where in the emotion, if we talk about pro-social in interpersonal behavior, in the emotion part, we have empathy, sympathy, compassion. In the cognitive part, we have what we think about others, our pro-social attitudes, kindness, pro-social values. And finally, in behavior, we have a long list of different behaviors such as nurturing, helping, and sharing. Is it one thing? Are they related? And how are they related? Do they represent shared environmental factors? Do they represent shared genetic factors? This is something that we were very interested in finding out. So for example, one study used questionnaire measures, and we asked parents to describe their children's pro-social behavior. We had different items measuring sharing, social concern, kindness, helping, and empathic concern. These were all 7-year-old twins. And we found a single common factor that accounted for most of the variance in most variables, with kindness as an exception. And then we said, okay, let's look at what happens with the twins. We looked at the correlations between twins. I don't need to represent the twin model anymore. And what we find is that for all the 5 variables, we have stronger correlations for monozygotic twins than for dizygotic twins. And there is one exception, which is kindness, where we see that the dizygotic correlation is also pretty high. And this is indeed the only of the 5 variables that has unique shared environmental variable variance. And the other variables have a meaningful genetic contribution of heritability running from 44% to 76%. And indeed, we can run a multivariate genetic model, and we can see that there's a common factor that is genetically influenced by 69%, and it loads on all of the items, all of the factors load on these higher order factors, and all of these factors also have their own unique genetic component and their own unique environmental component, and kindness has its own unique shared environment effect. So this is one set of findings. Another set of findings is to look at the motivation. When we say kindness, do we mean that kids care about others? What do they think about others? Do they want other people to feel well? And so we looked at the construct called values, and this is Schwartz's definition desirable abstract goals that are organized based on compatibilities and conflict, based on their underlying motivations, because there are different values with different motivations, and they are supposed to guide behavior. This is why they're interesting to us as students of moral development. They are supposed also to help people think about their values, but what we think is most important for our purposes is that values can guide behavior. And Schwartz has shown across hundreds of samples from all over the world, from Brazil to New Guinea, that values are organized in this two-dimensional structure where we have openness to change as compared to conservation values as one major dimension. I'm not going to be talking about it. And the other dimension contrasts self-transcendence values, universalism, benevolence, where you think about others, you care for others, as compared to achievement and power, which are values of self-enhancement, when you focus on your own interests. And until recently, until about 10 years ago, nobody knew how to measure values in children until Anna Doring's group came and they devised this wonderful measure of values in children, where children get these cartoons, and each cartoon has a picture that describes somebody thinking about a value, wanting something, desiring something, and there is a small caption below the picture, and then kids have to rank these items based on how they would like to be in their life. And what we find is that when you run multidimensional scaling of these items, we get exactly the same structure, or almost exactly the same structure that Schwartz has described for adults. We find it with kids. We even showed it at the age of 5, which is the youngest known to me. So there's something about values, about the structure. Children who choose rich and powerful as highly important are less likely to choose help others as highly important, and vice versa, because this is one dimension of self-enhancement, self-transcendence that relates to behavior. So does it relate to behavior? At least in theory, it should relate to behavior, because helping others is motivationally against being rich and powerful, and the other way around. But does it happen, of course, does it relate to individual differences in behavior? So what we did that in another study where we looked, we let children share candy, chocolate coins in this case, between themselves and another person. And we had this dictator game, decisions, where children could choose to divide the chocolate candy equally or not equally, giving more to the other person, giving less to the other person. It could be either costly or non-costly. And in a previous study, we showed that values related to prosocial behavior even at the age of 5. We ran this again with the twins in our study at the age of 7 and then at the age of 8 and 9. And these are the correlations between the 10 values on the x-axis and prosocial behavior in the candy giving. So this is a real behavior. This is costly behavior. Children actually see the candy taken from them if they share it. And what we see is exactly as you would expect based on the content of values, is that values of self-transcendence, thinking about others, especially universalism, relate positively to prosocial behavior, whereas values of self-enhancement, and especially power, relate negatively to prosocial behavior. The results are stronger when there is a cost to the behavior than when there is no cost to the behavior. This again replicates what we found with non-twin children. So we find that values have a meaningful association with prosocial behavior. And of course, the interesting question now is, do we see any genetic relatedness between the two variables? And here we can see the correlations in monozygotic and dizygotic twins. Monozygotic are in dark blue. And we can see that for both values and costly sharing, we find a positive correlation for both kinds of twins, but a stronger correlation for monozygotic twins, indicating a genetic effect. When we look at the cross-twin, cross-trait correlation, so these are the results that we define on the left. But when you look at the cross-twin, cross-trait correlation, what we find, this is on the right, we find that if we take one twin's values and the other twin's behavior, we find about the same correlation for monozygotic twins, as if it was the same child. But we find a much lower correlation for the dizygotic twin. After all, it's not the same child. And this is an indicator of a genetic relatedness between a genetic overlap between the values, between values and between prosocial behavior. And we can use this to run a multivariate genetic analysis with prosocial behavior at the age of 7 and 8 and 9, and values at these ages. And this is exactly what we find. We find a genetic effect of 21 to 44% on both prosocial behavior and values. And we find, of course, a non-shared environment effect, no evidence for a shared environment effect here. And we also find some stability in both prosocial behavior and values across an almost 2-year gap. What we don't find is a cross-lagged effect. So we didn't find any evidence for prosocial behavior affecting values or the other way around. Possibly we're not addressing the right age for that. Maybe something else is happening. But what we did find is that there was a genetic correlation of 0.70 or 0.74 between prosocial behavior and values at both ages, which tells us there's something genetic going on that drives the variance towards being either high on prosocial behavior and prosocial values or low on prosocial behavior and prosocial values. So a shared genetic factor is related to both behavior and values in the case of prosocial behavior and values. Let me just conclude. We find that different morality constructs interrelate positively within a domain. For example, when we talk about prosocial behavior, then positive emotions, prosocial positive emotions, prosocial values, and actual prosocial behavior tend to correlate positively. It would be interesting to increase this to other values. For example, values of self-direction could relate to behaviors of mastery. And another possibility is that the value system may serve as an organizing structure for the association between different behaviors. So behaviors that are based or related to the same values would be related at the phenotypic level in terms of behavior. Or the other way around, behaviors that are opposed to each other are related to opposed values. For example, universalism and power will be also negatively related in behavior. Although we have to remember that this overlap is only moderate. It calls for understanding of discrepancies. For example, what happens when your values don't fit behavior. Etiological differences. We saw, for example, that kindness is its own story. It's not the same as empathic concern, for example. What does it mean that there's a genetic overlap between one trait and values? Does it mean an underlying joint cause? Does it reflect a third variable, maybe some brain structures? This is all an open question that we'd like to answer in the next few years. Thank you very much. I'd like to thank my students and my funders. Thank you.
Thank you, Ariel Nafo, for your wonderful presentation. I think we don't have any questions yet from YouTube, so I will start with mine. I will first do a question to Emma, Professor Emma. You started the twin studies by mapping the number of twins in Brazil, and then went on to validate questionnaires that are important for twin research, such as the questionnaire to assess zygosity, for example. Now you have dedicated yourself to research on behavioral observations from a psychological perspective. When you thought about studying twins, what were the questions you had in mind to answer, and what have been the challenges to do so? Thank you.
Thank you, Tanya, for your question. In my opinion, twins provide a valuable source of information for psychological studies regarding genetic and environmental influences. Maybe there still are some misconceptions concerning the nature-nurture dichotomy. I think that we have the challenge of going beyond simple heritability studies. I personally was very interested in questions of emotion regulation, well-being. And reading a recent review article of 30 years of research on well-being, I found out that sociodemographic variables predict very little of the well-being indicators. The great contribution to explain individual differences is of temperament, and twin studies are so important to give us information about this. Though personally I was interested in these studies on emotion regulation, and well-being.
Thank you, Emma. I think we don't have any questions. I will do my question to Sylvia Corti. Okay, so you are starting the first twin register in Uruguay. Are there any other registers in South America other than Brazil and Uruguay? How do you see the perspective to extend these twin registers to other Latin countries? Do you already have any idea how to make this happen? And what are the challenges?
Well, hello. Thank you, Tanya, for your question. Well, I don't have any other. I don't have notice of any other twin registry in Latin America besides the University of Sao Paulo panel and this one that we are trying to initiate here at Uruguay. I think that is an amazing opportunity for opening a new perspective of research, because the intention of our registry would be to open it to many research. I know that in other countries there are registers that focuses on one aspect. German registry focuses on studies on inequality, but we are trying to integrate different disciplines for a multidisciplinary approach because of the multifaceted characteristic of twins studies. So I think that our principal challenge would be create that interest in other researchers, and we are yet in conversations with many of them. The professor of sociology, Matthias, is
I think we are having technical
hilarious
problems.
because when we met them,
Hello, Sylvia.
can you hear me?
Now I hear you. Yes. You're having technical issues.
Can you listen to me?
Now I can.
I was okay, telling you that we have met. We have met with Professor Nathan. It was hilarious because when we about
Sylvia,
training rates.
It's very, we can't follow you. I'm sorry. I think you're having technical issues. I'm so sorry. I really would like to hear you. Yeah, try
well, sorry. Okay. I was telling you that when we first met Dr. Matthias Nathan for asking him a collaboration with this register, he told us that we have stolen his research by doing he was right, because we are ethologists, but we are trying to this line of research in our country. So by integrating disciplines, we are sure that the register would be by the specialists that we must have, and with a different approach, we are sure that we could have a good beginning for studies in twins in Uruguay.
Thank you, Sylvia. Thank you. It was better now without the video. And now I have a question from Emma Otta to Ariel. Ariel Nafo, he has this twin register that has already 70 years, and it's very nice to see the data that you showed. It's very, very nice. So Emma is asking you, how is your registry organized? What advice do you give to us who are starting twin registers in Brazil and Uruguay?
Thank you for the question. First of all, just a small correction. It's 13 years. We started, the twins are 15 now. We started when they were 3. What kind of advice? I think my best advice to everybody is to do this as part of a group that is very multidisciplinary, that has broad interests, because in order to understand development well, we need to think about many things, from health to cognition to development to social skills and so on. So it would be better to have several people who have different fields of interest, different fields of expertise. For example, I'm collaborating with an obstetrician, and that really helped me with understanding everything about birth complications and so on that might be very relevant for twins. And so this is my best advice to start.
Sorry, I have another question to you. Ariel, considering that irritability estimates the degree of variation in a phenotypic trait in a population, if these studies were conducted in other cultural contexts and with another population, the results obtained could be different. How do you evaluate intercultural studies?
I think it's a wonderful question. I think when we talk about heritability, we always have to remember that it's context relevant and development relevant. So even within the same sample, we will find different heritability estimates in different ages, even if we use exactly the same measures, or maybe because we use exactly the same measures. So we have to keep that in mind. And that also means that when we compare across cultures, the context might be very, very different in economic terms, in social terms, and so on. So actually, I wouldn't generalize at all from one country to the other. I think the fact that, as we heard from Emma, from Professor Otta, that there is very little work in data meta-analysis from Brazil, one of the biggest countries in the world, just tells us how imbalanced twin research is in the world. And that's so great that you're doing this in Brazil. And I don't think that we can generalize from American studies to North American studies to Brazilian studies, for example.
Thank you. We are now having a collaboration. We are using your scale, and I think it will be very nice to study here in Brazil, the relationships twins. There's another question here from Valentina. Sylvia, what do you think citizen science can contribute in these times of COVID?
Can you?
Yes, I can hear you.
Can you hear me well? Yes. Unstable, but yes, Valentina, thank you for your question. I think that there are just the times for these scientific and citizen science could be done all over the world, and could be, yes, tell me.
No, it's because we didn't hear you, but now we are hearing you. Sorry, we're having technical issues. So, but you can continue it, and if we can't hear you, I will tell you again, okay?
I have here
I'm sorry, Sylvia.
so I hope
we
you
can't hear you.
Hear me. But yes, citizens are. Sorry.
I think she she's having technical problems. So I will ask Ariel Nafo. There's another question for you from Liz Dilala. Ariel, how do you conceptualize kindness as a different behavior? Do you think it's more influenced by worrying about how others will think about how you are behaving in public? I'm sorry, your microphone is off.
Sorry, I said that's a great question. Specifically, when we talked about kindness, we talked about a sort of interpersonal positive attitude. It's quite different from the other variables that we had there, like helping and sharing, that are more behavioral. So kindness is more about what the child thinks, or what the child wants, at least the way we conceptualize it in that paper.
Thank you, Professor Ariel. And now we have another question from Suemi Tokumaru. Thanks for the nice talks. I have a general questions. Are you aware of studies of investigating parental investments in twins? This is general, so it's to Ariel. Sylvio is not here anymore with us, but Emma as well could. Ariel and Emma. Emma, your microphone is off.
I would like to hear Ariel's opinion about this. We are planning a study of Isabella with parental investment in twins. But I think that Ariel has a line of research on this, and I would like to hear his opinion.
I'm not so sure I understood the question. Does it refer to parental investment theory and the more evolutionary aspects of parenting? I'm not aware of work on that. Of course, everything could be construed as such. So, for example, we have data, and we also have a published meta-analysis on parenting and how it affects, or how it relates to twins' behavior, and the genetic and environmental contributions to the parenting that children receive. So there's pretty much work on that, and we know that it varies across ages and slightly across measures, and there's some genetic effect, but some of the effects are actually parent-driven. We don't know much about fathers. I don't know exactly what else to say. I can refer you to two meta-analyses, one by Reut Avinun and myself, and one by Ashley Clark and Alex Burt, a very good paper to read on the topic, but not specifically on parental investment in the sense of evolutionary contributions.
Thank you, Ariel. I think she was, but I think it was a very nice answer in general. I think we don't have any more questions. Let me see if someone... Now, Sylvia came back. We could try, Sylvia, if you want to answer... I think if you want to answer your question that we did before, if you want to complete it, try now. I think it will be the... Better now, connection.
Yeah, so sorry, yes, it was a change of IP, I think. So was just now. Well, about citizen science, I think that is a wonderful tool in those times because we could connect with people online, and we also could have our field work, our field work done direct in the field, direct with our subjects, although we couldn't reach them. So I think it's a wonderful tool for these times of coronavirus.
Thank you, Sylvia. Now we heard you. It was thank you very much for your contribution. Let me see if there's another question. Do you have any questions, Sylvia or Emma or Ariel?
I would like to thank Ariel and Sylvia for accepting our invitation, and also the research cooperation with your group, Ariel and Sylvia, has been very interesting for us. So I expect that we can publish rapidly our manuscript together and with the translation of the of your scale, of your questionnaire that was being answered by the mothers. Do you think that we could make a version? This is being discussed with a colleague from Rio Grande do Sul, Professor Christian Zanon. The interest of translating the mother version or the father version of your questionnaire to a children version. So the children could give their opinion about their relation, and we could compare it with the father's and the mother's opinion. And these perceptions may not necessarily be the same. What do you think about?
I think it's a great idea. I think we know that parents agree, but it's not a perfect correlation. It's far from that. When fathers and mothers rate the twins' relationships, so that's one thing. And we also showed a correlation between self-reported closeness between the twins and parent-reported closeness longitudinally. So there is some agreement on that, but it's far from perfect. And I think everything about relationship is what it is and also how it's perceived. So a study where we have different reporters on the same relationship might be very much telling. I mean, especially if you have a discrepancy between how twins perceive themselves. For example, one of the twins think they have a very close relationship with the other one, and the other one doesn't think so. That could be very meaningful clinically, for example. And so that's something you can benefit from when you are looking at child report or adolescent report on their relationship without specifically relying on the parents, because the parents see the relationships as very, very reciprocal, very symmetric, for the dimension of dominance, where there's no correlation between the dominance of the two twins. So either one is dominant or the other one is dominant. But closeness, conflict, dependency, and rivalry are considered very, very reciprocal. I'm not sure it's the same when the twins think about it themselves. So I think it's a great idea for a study.
Thank you, Ariel. Sylvia, would you like to, do you have any questions? Or Ariel Nafo, if you want to, if you have any questions to Emma and Sylvia.
Yes, I was very interested in that research of you about the values that guide behaviors. No? I think that is very interesting to make some research about that, because values could change because of culture. Culturally, could be different in different countries. So it would be very interesting to make research in different countries with different cultures about those values and about how those different values guide more or less that behaviors.
I agree. I think when we talk about pro-social behavior, things that are supposedly the same, like sharing candy, right? It's candy, it's the same thing, but it's not the same thing depending on what cultures' norms are. So are you expected to share? Are you expected to share just a little bit or not? That, of course, is affected by the values of that culture. So the behavior of the children that we're observing is always culture dependent. And of course, when we do a twin, we usually don't really mind because we're interested in the within culture variability. But some of the variability is between cultures. And I don't know about many studies that try to incorporate the two designs, like the cross-cultural design and the behavioral genetic design in the same model. I think it's really hard to do it because of many reasons, including culture and comparability of measures. And, even if we were to do a study, then our twins are not the same age as your twins, for example. So that complicates things. But ideally, that should have been done in order to try to figure out how culture affects individual differences, as well as in the interaction. Is, for example, the heritability of pro-social behavior different in a country where children are always expected to share and country where children are not always expected to share? Probably it's different, but it's work that has to be done.
Thank you. Thank you very much.
Thank you, Ariel. Let me see if there's another question. I think we finished. So I will thank you, all of your three presentations. It was very nice to be here with you. And now we will have the second session. That will be the statistical analysis of twin. And I will call Professor Claudio Possani. Please, welcome.
Thank you, Tanya. Well, let's continue our event. It's a pleasure to be here. And the name of this session is Statistical Analysis of Twin Data. It's a very interesting subject. And it's really a pleasure to be here with Vinicius Fraser Davi, who is a very special research. You'll see in his talk what I'm talking about. But let me say something before. I am a mathematician, and I've been working with mathematics for the last 40 years. Sometimes I worked in pure mathematical problems, but I have a lot of works with people from different areas in what we call applied mathematics. And what's the more easy to see is that when we want to apply mathematics to a biological or social problem, people from the humanities or biological sciences don't understand mathematics. They don't want to study mathematics. And we, mathematicians, have to learn a little bit about their subjects, and we work together in a very interdisciplinary way, but none of us are autonomous in the area, because in my experience, I didn't understand in a deep way what was going on in biology or in a social or economic problem. And my partners also didn't understand in a deep way the mathematical tools we were using. Well, it's a way of working. But with Davi, something different happened, and you'll see Vinicius in his talk. He's not a mathematician in his basic formation, But he started studying mathematics in a very hard way. And he got an autonomy. He's a very autonomous in statistics. And that's quite different, because although he likes to work in teams, I have this experience, we work together, he likes partnerships. He likes doing things together with other people from different point of views. He accepts very well interdisciplinarity, but he's very autonomous in what he's doing. And that's something that's not so common. David is a special research in this way. I've found so few people that came from another not exact area and who studied with in such a hard way the mathematical tools and could be so autonomous. So welcome, Vinicius. It's a pleasure to be here with you. And all of you, you understand what I'm talking about when you see the talk of Vinicius. Well, the name of his talk is Mixed Models and Statistical Analysis of Twin Data. And, well, the best person to talk about that is Vinicius. Thank you, Vinicius.
Thank you, Claudio. Thank you. Isabella, I think you can start.
So, good morning, everyone. My name is Vinicius Frais David, and I'm a researcher in the Institute of Psychology of the University of São Paulo. I am a psychologist, but I have been working with statistics for some years. And today I will talk to you a little bit about the use of mixed models to analyze twin data. I will use almost no mathematics. The idea here is to show what it can do more than how it works. Usually when I talk about statistics with other researchers, most of them identify it as its purpose, which is to teach people to use a limited sample to make intelligent and accurate conclusions about a greater population. In this sense, statistics is viewed as a tool, as a mean to an end. But statistics is also a field of knowledge in constant change, and we have to keep track of the new developments that can help us in our studies. And what are the issues with twins designs? Probably the simplest design that we can have is the one we want to compare two groups in a particular trace. This is what we have. Several subjects in group A, each one with observed trace, and another group B with its subjects and its observed traces. This is simple, and we can compare many characteristics of the distribution of these traces between groups. When you have to do that with twins, at first glance, it doesn't seem very different. We still have two sets of subjects and its observed traces. The problem is that these are not independent subjects as they were in the first case. Here we have pairs, pair 1-1 and 1-2, pair 2-1 and 2-2, and so on. And as they are pairs, we could expect them to have some covariance. More than that, we are very interested in the value of this covariance. After all, if we find that, let's say, monozygotic twins have a bigger covariance than dizygotic, we can suspect there are some genetic influence on this observed trace. Here's an example. If I measure the heights of monozygotic pairs and dizygotic pairs, we can see that the monozygotic heights are much more similar between siblings than dizygotic ones. And so there's probably a genetic influence on this trace. We know there is, in this case, this is just an illustrative example. And what do I mean when I say genetic influence? Well, we know that heritability is defined as the fraction of phenotype variability that can be attributed to genetic variation. We can illustrate that with the simple formula. It's a proportion of two variances. We also know that The most used model in heritability studies is the ADCE model. It divides the factors that can influence a person's traits between 3 genetic factors and 2 environmental ones. So, we have an additive, dominance, and epistasis genetic effects, and then we have common environment that are experienced by both twins, and unique environment that is experienced by each one differently. Using the same formula that indicates the variation of each of these factors, we can now describe heritability as the variance of additive, dominance, and epistasis effect over the total variance of a trace. We can use submodels of ADCE model too, if we exclude some of the factors. One of the most common is the ACE model, or ACE model, in which we consider all the genetic variation to be an additive effect. So, we are interested in variances and covariances. How can we calculate these covariances that we are so interested in? 2 of the most used techniques are the intraclass correlation coefficient and the structural equations modeling. The ICC quantified the degree to which individuals from a fixed degree of relatedness resemble each other. It's interpretable as a common Pearson correlation from -1 to +1. The biggest difference to a regular Pearson correlation is that the order of the pair is not important. What is what we need? Because usually there is not a good reason to select a twin as number 1 or number 2. The structural equations modeling is the most common model, have been the most common model in twin studies for a long period. It's a very versatile model. It uses several regressions that can be calculated at the same time, and there's the possibility to use observed and latent variables at the same time. Although these are interesting approaches to use in our studies, they have some limitations. For the ICC, the most notable limitation is that it compares only 2 subsets, for example, monozygotic siblings or dizygotic siblings. If you have several subsets like zygosity, gender, age, etc., several analyses will be needed. For gender, you have to calculate one index for male monozygotics, other for female monozygotics, then male dizygotics, and then female dizygotics, and so on. It increases the chance of type 1 error. And of course, you need a larger sample size to do that. For structural equation modeling, we know that most of the procedures that have been suggested involve known standards and complex model specifications that are challenging for the average user and thus susceptible to errors. Convergence problems have been observed in some procedures, so maybe it doesn't work properly for you, for your study. Some of the most promising approaches are not easily available in conventional structural equation modeling softwares. And again, we have this problem of sample sizes. When you can make a sample size calculation, but usually the rules of thumbs is that you need more than 25 observations per parameter. So we get to the mixed models as an alternative. The main difference of mixed models to others, like the regular analysis of variance, is that mixed models include a random effect to our fixed effects. A random effect supposes that the data come from a hierarchy of different populations and the differences are related to this hierarchy. It has the assumption that individual traces are not related only to independent variables. There is the presence of a known random error. It is very used in educational studies. Let's say we want to compare results of a test from children of private or public schools. And we have data from some schools of each group. The nature of the school is our fixed factor, private or public. But we have to consider the school itself as a random factor. After all, we expect to have some covariance due to the school, and not only for its nature. Some schools will have better facilities, better teachers, and so on, despite the fact that they are public or private. And this logic have been adapted for twins designs. We have some fixed factors, but we also expect the individual traces of our subjects to be related to the trace of the other sibling. So, in twins designs, we use the pair of twins as our random factor, and now we can calculate the covariance among them, and consider that in a more complex analysis. The total variance of any mixed model is the sum of the random factors and the residual variance. So here will be the variance of the pairs plus the residual variance. Which leads us to a second problem for twins designs. The first one was to deal with the pair covariances, and it's similar to many other studies. It's not much different to the school example I used before. But when we use school as a random factor, for example, we can assume some kind of regular distribution among schools, and so we can use only school as a unique random factor. But with twins, we want to calculate different and very specific covariances to investigate to which extent our traces can be considered heritable. We need to know at least two covariances, one for the monozygotics and other one for the dizygotics. These are just some examples to illustrate how the possible difference in the covariances can lead to different conclusions. It's not very important what is in the table, but just to show that it's important to know the two covariations. So, what do we have to do? We need to separate dizygotic covariance from monozygotic ones. And recently there was developed a way to do that using mixed models. It can be done by separating a covariance that is function of the pair of twins, regarding the fact that they are monozygotic or dizygotic, from the extra covariance that is due to the pair be a monozygotic pair. In other words, the difference between the covariation of monozygotic and dizygotic pairs. So now, our total variance will be the variance of the pairs plus the extra variance of monozygotic pairs plus the residual variance. And how do we do it? I'll try to show you with an example using Stata, but it can be done in R, SAS, or even SPSS using the appropriate comments. The first thing you need to do is organize your data set. Each subject must be in a different row, and because of that, we will need a variable identifying each pair. You can use any number you want as long as it's the same for each pair and each pair has a different number. Then you can have your variables of interest, like zygosity, gender, or any other. This is no different from any other analysis. The trick is to create these additional variables. They will be responsible to separate the monozygotic covariation from the dizygotic. Firstly, we will identify your subject as being monozygotic or dizygotic. The easiest way is to put zeros for dizygotic and ones for monozygotic twins. Remembering, you have to declare it for every subject, even knowing that the sibling will have the same number here. Then you create two new variables that I'm calling here DZ1 and DZ2. You have to assign each of the dizygotic twins from each pair to one column using zeros and ones. The first twin is 1 and 0, and the second is 0 and 1. It doesn't matter which one is which, as long as they are assigned differently. Monozygotics will only have zeros here since they were already defined in the previous column. And then we have our comments. Here we have two examples that can be modified to fit different designs. You declare that you're using a mixed model. Then you have your observed trace, and then the fixed factors. Here I used only male. That is my gender variable. And here there is a more complete model. The most important part is here, when you declare your two random effects. The first one is the pair random effect. Independently if it is monozygotic or dizygotic, it will calculate the covariance of the pair that are common to monozygotic and dizygotic twins. The other one is only valid for monozygotic. It will calculate the difference of covariance between monozygotic and dizygotic pairs. Then you can specify the structure for the covariance matrix of the twins. Usually you can consider it to be identity. The main difference between these two commands is that on the first one it uses maximum likelihood estimation with a chi-square distribution. And the second one uses a restrictive maximum likelihood estimation with a t distribution. As a rule of thumb, if you don't have a better information to choose between them, if your sample is small, use the restrictive. If it's large, you can use the maximum likelihood. And then we can look at the outputs. The first one is very like any other analysis. You have your estimate there, your errors, your statistics, values, p-values for each factor, and you say that it's just the same. The interesting part is this other table, where you can find the values of our variances. The first one is the part of the variance due to the covariance of the pairs. Here we can see how the pairs of siblings are close to each other regarding the zygosity. The second one is the increase of variance explained by the covariance for being monozygotic. This is only valid for monozygotic twins. And the third one is our residual covariance, the portion of the variance that is not explained by the fact that they are siblings, mono or dizygotics. Remembering the previous formula, the total variance is the variance of the pair plus the extra variance of the monozygotic plus the residual variance. Now we have some values. And what is the common variance of the pair? Well, if we are using an ACE model, what is common for every pair regarding zygosity? We have at least half of the genetic similarity and the common environment. I'm also assuming here that the twins were raised together. This is the extra monozygotic variance. The monozygotic covariance minus the dizygotic covariance. Since monozygotic are expected to have the double of the genetic similarity when compared to dizygotic, we can expect this value to be half of this effect. So it's half of the additive effect. And the residual variance is what's not explained neither by the additive effect nor the common environment, if we are using an ACE model, so we can assume it's the unique environment effect. Putting in numbers, we can conclude that 15.6% of this trace variation is due to common environment variations, 60.7% is due to additive effects, and the unique environmental variation is responsible for the 23.6% left. Now, I have to say that mixed models also have some limitations. First, show you how to perform it with an ACE models. And we know that this model can overestimate the genetic effects, so we need to be careful in the sense. It is possible to include dominance and epistasis effects, but this gets much more complicated and lost the advantage of being a simple model. The way I show you, I only analyze one independent variable at a time. But you can do it with several ones. That's not that hard. You just need to include another random factor that will be the subject itself. I also use a linear model, but you can use generalized mixed models that deal with different distribution like binary, ordinal, and so on. Finally, as far as I know, if you have both observed and latent variables as independent variables, there is not a solution yet for mixed models. This is a real limitation, and thank you.
Thank you, Vinicius. Very, very beautiful exposition. Thank you very much. I would like to say, to start with, I would not like to make you a question or put you a question. I would like to ask you, let's change the schedule and you give another talk with 10 times more, and you explain us everything you did in a very more detailed way. Very, very, very interesting. Vinicius, this concept of heritability, is it too old, new? Because I have never heard it before. I heard it for the first time in a talk discussing it with you. And how long people are considering this concept in psychological studies, and how disseminate it is in the community of psychologists, how clear this idea is to research in, for instance, twin studies or behavioral studies.
Well, a very hard question. I'd say it has been there for some decades now. Plumbing, I guess, in 2019, published a book, Blueprint, How Genetics, something. It's a very interesting book for people that don't know very much about the concept of heritability, because it summarizes his research for the last decades. And if I'm not wrong, something like 40 years of research. So it has been there for some decades now. How disseminate? I can say a lot about other countries. I would say that in Brazil, it's not very disseminated yet. If we look at the disciplines of different psychology institutes of colleges, you won't find very much about that. I think in Institute of Psychology, we don't have a single discipline that discuss that with undergrad students, and I think that's not good. We have to keep track of that. I really think behavior genetics is something that we can't be so unaware of that as a psychologist. I have never heard of it until some years ago, and I've been working with research for more than 15 years. I am a psychologist. I had in my graduation or even in my master's, I never heard about it. There was not a single professor in University of São Paulo of Ribeirão Preto, that's where I was graduated, that was aware of that. Nobody told me. So I think in Brazil we have a long way to go. Yeah, but the concept is there for some decades now. Professor Turkheimer will talk something for us on the next session about the use of behavioral genetics in sociology. And I understand your question, because really, as a psychologist, I was not aware of that until some years ago. But the concept is there. In Brazil, we have a long way to go.
Yeah, because it's interesting. We are acquainted of thinking about something more simple. Well, I have some genetic factor to have some disease or to have some characteristic, my height, the color of my eyes, it doesn't matter. I have some genetics reasons to have some characteristics. And so, well, there is a probability of having or not having that characteristic. Maybe I have the tendency to have some disease, but maybe I will not have. But it's a very simplified way of thinking. And when you put in terms of heritability, you change the way of you are thinking about this subject. If you have in mind some other examples, I think it would be very helpful for people who are attending us to have another examples of how this point of view can change the way we think about some characteristics or some behavioral characteristics.
Yeah, something we always have to keep in mind, especially when we are talking to people that are not from this field, is that we are not talking about what is genetic or what is environmental. Everything is genetic, everything is environmental. My example, right. If you are a neurotic, you can't, and I say, 40% of your neuroticism is because of genetic. You can be neurotic if there's not an environment. Everything is 100% genetic and 100% environmental. It's always something, we are in an environment, so we can't separate one thing from the other. What we try to do when we calculate heritability is, and I use that word more than once in my talk, variances. We are talking about variances and covariances. So considering the variance of this trait in a population, how much of this variance can be attributed to a variance of the genetic or a variance of the environment? So, and that's why in the last lecture, I think when Tena asked Ariel, he said that can be different from population to population, from different cultures and from different periods of time, probably. We have to keep that in mind, especially when we're talking, especially when we are talking with people that are not from the field, because usually when we talk about that, some people, no, that's not genetic, that's environmental. No, it is environmental, of course. But we are talking about the variation. And it varies a lot from population to population. If the population is too similar in context aspects, probably the genetic variance will be more important to explain the variance of any trait. And but we also, you asked for examples, every trait has some, the variance of any trait has some cause in the genetic variation. That's I think it's the first law of behavioral genetics. Every trait. Think about something, anything that you can think about as a human characteristic. It has some influence, is influenced by some genetic variation. Everything.
Well, I have a question. How is the minimal sample size to conduct such analysis? How many pairs are necessary? Could you talk a little bit about that in your talk, but could you say some more words about that?
Yeah, calculating sample size is never too easy. We have some rules of thumbs, but it can vary a lot. It can vary a lot from trait to trait, because you have to look at the distribution of your sample. If we have a regular distribution, you're probably it's like that thing with 30 people per group. That's not always true. But what I can say is that you need less. A smaller sample size to use a mixed model than you need to do a structural equation modeling. And I'm not advocating against structural equations modeling. The question is that if sometimes you try to use a too powerful tool and you have to pay a price for that. If your question is not that hard, if your question is more simple than that, then you can use a simpler model. The most powerful model are not always the best model to use. You can use more simple models, and because of that, you need a smaller sample. You have sometimes more power, estimate power. Again, it's hard. Unfortunately, I can't give too much details on that because it's very, very, very complicated. Calculating sample size and power for this kind of model is not as easy as calculating that for a regular ANOVA or a t-test or something like that. It involves some more hard things, but it can be done. It can be done. I just don't have the time to explain here. I would need a board and several mathematical formulas to explain. But we can calculate the sample size and the power.
Well, Christian Zanon is asking another question following the first one he asked. How do you estimate the power? Can you tell us a little bit about that? How can you estimate this power?
Well, that's what I would say. You need some more complicated modules. You have modules to estimate power in R. In Stata, I'm not sure, but probably you have. But again, it's hard to explain. It's really hard to explain here. I have to say I'm sorry for that, but it's not easy to explain here in 1 or 2 minutes. If you can, you have my email. You can email me, and I'll send you. If you try to read our papers about it, you see a lot of mathematical formulas. It's very complicated. I tried to keep it simple here to show you what it can do and how it can be done. But this kind of detail are not that easy. But again, about sample size, you can the bigger the sample size, the more power and the better things. But it's hard. Sorry.
No problem, Vinicius. You have been so clear so far. Well, I would like to thank you, Vinicius. It was a very beautiful talk, as I told you. And as I told you also, I would ask you to give us a course on that, not only a small talk, but it was very interesting and very challenging. And what I like the most is that you are talking about something. It's not quite new, as you said, but it is new for us. It's something that we have to put in our mind. It's a new way of thinking that we should, how we are doing research in the subject, to start thinking in such a new way. And I loved it so much in your talk. So Vinicius, thank you very much. It was really a nice talk. Really a pleasure to be here with you. And at the same time that I'm saying thank you, I am, Tanya is here, and you will be the host of the next part of our event. So it's very easy to change. Thank you and hello. You are the guy in charge from now on. Thank you, Vinicius.
Thank you all. It was very, very, very good. And now I have the pleasure to announce our next speakers. Our next session is called Subjective Well-Being and Individual Differences. And our first guest is Professor Marco Pluis. He's PhD, Chartered Psychology and Associate Professor in Developmental Psychology at the Department of Biological and Experimental Psychology at the School of Biological and Chemical Sciences, Queen Mary University of London. He's also a visiting fellow at the London School of Economics. His research deals with questions related to developmental plasticity, genetic environmental interaction, and positive development. More specifically, Dr. Pluis investigates the individual differences in the capacity for such developmental plasticity as a function of different individual characteristics. His talk is called Environmental Sensitivity and Vantage Sensitivity I welcome Professor Pluis. Thank you for your participation.
Thanks a lot. So let me just get my slides up. Okay. Can you see my slides?
Yes.
Okay, great. So thanks a lot for the invitation. I've never been in Brazil, and I'm a little bit in Brazil now, I guess, with this talk. I have a postdoc that joined me who is from Sao Paulo, so it's I feel a little bit of a connection to Brazil, and maybe one day I'll be able to visit. So I'll talk about environmental sensitivity and vantage sensitivity, and let me get started with defining what I mean with environmental sensitivity. So environmental sensitivity is really a very fundamental trait that we can observe in humans, but also many other species. It is defined as the simple ability to perceive and process information about the environment. So we all function within a context. We live on the planet Earth. We live on a specific continent in a specific setting that has challenges and opportunities. And environmental sensitivity allows us to read the environment, perceive differences in the environment, specific aspects of the environment, process that information, and then act upon that. So it's really absolutely important. For example, environmental sensitivity allows us to perceive threats, to perceive potential adverse situations and then prepare and respond to those. But it's not just helpful in order to perceive threats, but also to be aware of opportunities, such as an oasis in a desert. So environmental sensitivity helps us to perceive information about our surroundings, process them in order then to respond to that. So it's really important for us to be sensitive. Anyone that is not sensitive would struggle. So therefore, do we expect that everyone has the same degree of environmental sensitivity, or might people differ in their sensitivity? Well, some evidence that people might differ in their sensitivity is provided in many psychological concepts, which when we look at different markers of sensitivity, such as specific personality traits or stress reactivity, which we consider markers of sensitivity, we do see that people differ in those, that some people have a higher stress reactivity, other people have a lower stress reactivity. They're more affected by their experiences or less affected. So therefore, the hypothesis that we work with is that some people are generally more and some people are generally less sensitive to what they experience. And this is what we've proposed, or what I proposed in that paper, but there are many theories that are focused on that observation. So let me try to summarize that, the different theories. Most of individual differences in response to sensitivity have been framed within a diathesis stress concept. According to this diathesis stress concept, two individuals may not differ in the absence of a negative influence. Their differences emerge in response to a negative influence, with some people being resilient, meaning they're not really negatively affected by the negative experience, and some people being vulnerable, so they show a decrease, for example, in their well-being or in any level aspect of their functioning. So diathesis stress or vulnerability stress usually focuses on vulnerability in response to adversity. And that a lot of the research that looked at individual differences used that model. And then more recently, about 10 years ago, or maybe even 15 years ago, differential susceptibility has been proposed as an alternative model to look at individual differences. And according to differential susceptibility, it's not so much that people are resilient or vulnerable, but maybe more that the resilient people have a low degree of sensitivity and the vulnerable ones have a high degree of sensitivity. And that sensitivity is not just restricted to the experience of adverse adversity, but also in response to positive experiences. In other words, if those two individuals would now experience something positive and they are different in their sensitivity, we would expect that the individual with low sensitivity would also benefit less from a positive influence, and that the individual with a high sensitivity, who is more negatively affected by adverse conditions, would, in response to positive influence, actually show an increase and benefit significantly more. Therefore, factors that are associated with both a heightened response to negative and a heightened response to positive experiences should be considered sensitivity factors rather than vulnerability factors. So we basically have two sides to this story of differential susceptibility. We have individual differences in response to negative experiences that is captured by the diathesis stress model. We do have lots of suitable terminology and lots of research that provides evidence for that. What was really new by the differential susceptibility model was the idea that people also differ in response to positive experiences. And we didn't really have terminology to describe that. And that's why we proposed the terminology of vantage sensitivity to describe this positive side to differential susceptibility. So according to vantage sensitivity, two people may not necessarily differ in the absence of a positive influence, but it is in response to a positive influence that we see those differences emerge. With some benefiting from the positive influence, they show vantage sensitivity, and some not benefiting, they show vantage resistance as a function of vantage resistance factors or vantage sensitivity factors. So basically, these are all models of environmental sensitivity, and vantage sensitivity is the one that describes individual differences to positive experiences. And let me just give you one empirical example. So this is a study where we measured sensitivity in 3-year-old children using an observer rating system. So this is not parent report or self-report. This was rated by psychologists and was mostly done by my colleague Francesca Leonetti. So we had a sample of almost 300 children, and we did have measures of parenting, that sensitivity measure, and we looked, in this particular example, we looked at the outcome of social competence. And what we found is that there was an interaction that fit best with vantage sensitivity rather than differential susceptibility or diathesis stress. And let me just show you the findings here. So we do have authoritative parenting at age 3, that was based on parent report, and we have social competence of the children at age 3 also based on parent report. And here are the two, for the follow-up analysis, we created extreme groups of high sensitivity and low sensitivity. And the low sensitive children, for them really, there was no association between authoritative parenting, a positive parenting style, and social competence. But for the children that had high sensitivity, they had higher social competence when authoritative parenting was higher. So those differences emerge particularly in a context of positive parenting. That's why these findings are more consistent with vantage sensitivity. Next, I'd like to talk a bit about the mechanisms of environmental sensitivity. Why are some people, some children and adults, more sensitive to what they experience? Well, most of the authors in different theories that have been proposed on sensitivity suggest that sensitive individuals have a more sensitive central nervous system on which experiences register more easily and more deeply. There are various factors that have been associated with sensitivity. There are various genetic factors, physiological factors, and psychological factors, but they all really are connected with each other, and they seem to reflect or contribute to this heightened sensitivity in the central nervous system. And there are various brain regions that might play a role here. But one that seemed to be of particular interest is the amygdala. The amygdala is a very central part of the brain. It's part of the limbic system, had a primary role in the processing on memory of emotional reactions, not just negative ones, but also positive ones. And interestingly, several of the genetic factors of the candidate genes that have been associated with sensitivity also seem to be associated with amygdala reactivity, so the function of the amygdala. And let me just give you one example with the serotonin transporter polymorphism. So there are basically, this is the serotonin transporter is a gene, and there is a polymorphism in that some people have a long version and some people have a short version. And what they found is that people with the short version of the gene, they have a higher amygdala reactivity. That means the amygdala is more responsive when challenged. In this particular sample, it was looking at people's faces that show negative emotion. We also looked at amygdala volume in a very recent study, which should come out very soon. So in this study, we looked at the size of the volume, the size of the amygdala, but also the hippocampus, but we didn't find anything with the hippocampus. We did find interactions with the amygdala here in red. So basically what we did in this study, and this is a subsample from the TED study, we looked at environmental quality in the first 9 years of life. We then had measures of the amygdala volume, which were collected between the ages of 10 and 12. And then we had teacher-reported total problems of these children at age 12. And that was a sample of about 60 children. What we found was that there was a significant interaction with the left amygdala volume. And we followed that up with these simple slopes. And as you can see here, so we have environmental quality across childhood, and we have total problems at age 12. And then we have the children with small amygdala and the children with large amygdala. These are all boys only. What we can see is that for the ones with a small amygdala, there was no significant association between the quality of the environment across the first 9 years and the total problems at age 12. But a significant correlation, association, was there for the children with a large amygdala. And the shaded area here is the regions of significance. So we tested at what level of the environmental quality does the association become significant. And it was really at the higher end, at the high quality of the environment where this interaction became significant. In other words, children with a large amygdala are particularly responsive to higher quality. So this finding was more consistent, again, with vantage sensitivity. Maybe important to say that the environmental quality scale here was really capturing repeated measures of the parenting, parental involvement, but also socioeconomic factors, the material environment of the child. I want to just share one slide on the genetics of sensitivity. So much of the research in the last 10, 15 years featured a candidate gene approach where people focus on individual genes. And many of those gene-environment interaction findings were consistent with different patterns of environmental sensitivity. But more recently, people have been adopting or understood that complex traits such as sensitivity are not the function of individual genes, but more likely the result of many different gene variants. So we're talking about thousands of different gene variants. And this is a very interesting study, the first study that captured that by Rob Kears and Associates. So they created a polygenic score based on discordant monozygotic twins, quite an interesting and powerful approach. And based on that, they then tested in a separate sample of almost 1,500 children, where the children that have more of those sensitivity genes, whether they are more affected by both negative and positive parenting. And that's indeed what they found. They found a significant interaction between their polygenic score of sensitivity that included more than 25,000 different gene variants and parenting in the prediction of emotional symptoms. So here is the figure from that paper. So at the bottom, you see the standardized polygenic score for sensitivity. So at the left here, you have the people that have low genetic sensitivity. And here you have the children that have high genetic sensitivity. And here are the emotional symptoms. I think that's measured with the SDQ scale as well. And what you can see is at the lower end of genetic sensitivity, the children didn't differ regarding parenting and emotional symptoms. But the more genetically sensitive children were, the more they were differentially affected by parenting quality. In that, highly genetic children that experienced negative parenting had higher emotional symptoms, higher emotional problems, but genetically sensitive children that experienced positive parenting had the lowest problems of all. So a finding consistent with differential susceptibility. So all of this has been combined in the neurosensitivity hypothesis, which suggests that we do have many different genes that contribute to structures and functional aspects of the central nervous system that then contribute or drive this sensitivity. And in response to specific environmental influences, that could be specific negative or positive influences, we do observe specific behavioral aspects like psychological aspects or physiological aspects that have been associated with sensitivity. So the neurosensitivity brings that all together, suggests that yes, there is a genetic contribution. Most likely that will be associated with specific aspects of the central nervous system. But it's very complicated measuring the brain, and it's expensive, and it's also complicated measuring genes. And it's actually, at this stage, not possible, based on genome-wide data, to really predict an individual's sensitivity. So it's still very helpful to use phenotypes of environmental sensitivity. And the one I want to raise here is the one that has been proposed by Elaine Aron, actually in the mid-90s already, in her work on sensory processing sensitivity, which is one of the leading theories on environmental sensitivity. So she suggested that sensory processing sensitivity is a personality trait that is fairly common, and that is characterized by people having a heightened sensory sensitivity, but also to be more aware of subtleties in their surroundings. These people process experiences more deeply and are more easily overwhelmed in a highly stimulating environment. These are just a few of the aspects that describe their personality trait. So she created the highly sensitive person scale. The original scale included 27 items and was designed for adults. And over the last few years, we created brief versions for children as well as for adults, with 12 items each. And I'll show here the scale that we use for children. So these are 12 items. So that is very much based on the adult version. In the meantime, we've just got a paper accepted where we included some additional items to strengthen the psychometric properties. But we've used this scale now in many different studies and found fairly consistent findings. So let me just plug as well the online test. So this year we published a website, sensitivityresearch.com. And on that website, you will find information about sensitivity, frequently asked questions. You can link up with other researchers that do research in that area. You can sign up and become a free member, where you get access to all our measures for free. There's also blogs where we once or twice a month present new findings in a format that is also suitable for the general public. And when you click on sensitivity tests, you can choose between the 3 different tests that we have there. And here's just an example for the adult test. So this is based on the research measures that we use. This is the 12-item measure for adults. We also have the one for children, self-report, and we have the one for children based on parent report. So if you go on the website, you can do all of that and find out about your own sensitivity. Given that this, obviously, many of you will be very involved in twin studies, we've conducted and published a study on the heritability of environmental sensitivity using this scale, the highly sensitive child scale, so the HSC scale. And we used a subsample of the twins early development study, which I assume most of you will be familiar with, which is the largest twin study, one of the largest twin studies in the UK. And we had data from about almost 3,000 twin siblings, and we conducted the study when they were 17 years of age. And what we found is that we had a heritability of about 47%, which means that 47% of the differences in sensitivity. So a substantial heritability of sensitivity, but fairly similar to other personality traits. That suggests that there is scope as well to explore the genetic components in more depth, and for the genetic studies using genome-wide approaches, we will need very large samples, and that will be a bit of a challenge. There's also quite a bit of work that has been done on sensitivity groups. There have been books that have been published, particularly in the general domain for the general public, popular psychology. And the metaphor that has been picked up by many people is that people fall into two groups. That we have the dandelions, that's the majority of people, about 80%, they are generally less sensitive. And then we have the orchids, these are the highly sensitive ones, they make up about 20% of the population. Now, whenever I used sensitivity scales, whether for children or adults, I often found a normal distribution. So I've doubted whether those groups actually existed. And the proposition that those two groups existed were not really based on strong empirical evidence, at least not in humans. So we thought we need to figure out whether that's actually the case. So we tested in our data whether we do find groups. So first of all, we looked at the distribution, and we did find sort of a normal distribution. That's what we generally find. But then we applied a latent class analysis to our samples. And we did that now in many different samples. And we often found this following solution. We do find that this normal distribution can be differentiated, can be further divided into three different groups. We do basically find 30% at the lower end, 30% at the higher end, and 40% are sort of in the middle. And we did find that in undergrad students, we did find that in children, we did find that in adolescents, we did find that in samples in the UK, in the US, and other cultures as well. The proportions change, but on average, we basically find 30% at the bottom, 40% in the middle, and 30% at the high end. So basically, we do find evidence for dandelions. They make up about 30% of the population, not 80%. And we do find evidence as well for the orchids. Again, they make up 30% rather than 20% of the population. And then we have the middle group, and we had to find a suitable flower for that. And we thought that tulips might be the right term for that. So if you go on the website and you do the test for sensitivity, it will give you the mean score of our sensitivity measure, but it will also tell you whether you are a dandelion, a tulip, or an orchid. So I'm coming to the end of my talk. So I'd like to conclude with and summarize and suggest that people generally differ in their environmental sensitivity, even though environmental sensitivity is such an important trait for every single individual. People differ in their level of sensitivity, with some being more and some being less sensitive. And that has been associated with different genetic, physiological, and psychological factors. All of those most likely reflect a heightened sensitivity that is driven by the central nervous system. And there might be specific brain regions. I mentioned the amygdala as one example. There are other brain regions that have been identified. And all of this suggests that we should expect, whether we look at the effects of positive experiences or negative experience, we should expect that people differ substantially in how they respond to them because they differ in their environmental sensitivity. And that concludes my talk. So thank you very much for your attention.
Thank you, Professor. Thank you. Wonderful talk on a very interesting topic. Thank you for your talk. We'll open to questions, but first, I want to call our next speaker. That is Professor Eric Turkheimer. He is the Hugh Scott Hamilton Professor of Psychology at the University of Virginia. He studies the genetics of complex behavior using empirical, quantitative, and philosophical methods. He is also interested in individual differences in personality, classification of mental disorders, and philosophy of psychology. His lab studies how interactions between genes and environments shapes the development of many aspects of the human behavior. And his talk is called Behavior Genetic and Mainstream Social Science. Welcome, Professor. You can start a talk. Oh, I guess it's Isabella. Isabella can start his talk. Thank you.
Hi, everybody. It's nice to be here, or it would be nice to be here if I was actually there instead of here up in my attic. And oddly, though, I've spent the last 8 months continuously on Zoom. It feels like it's the first time I've recorded a talk. Ones I've done before, I've done live. So I hope you bear with me, and I hope I can get this right. So let's see. Next step is sharing my screen. And there we go. What I'm going to talk about today is the relationship between behavior genetics generally, and particularly twins, and what I call mainstream social science. And I'm happy to come to a twin conference, but what I have to say mostly I think applies to modern DNA-based genomics just as much as it applies to twins. And that's kind of an interesting topic that I probably won't have time for. And it kind of starts with my own intellectual history with this stuff. I've been a skeptic in many ways about the meaningfulness of the outcome of twin studies for a long time. If you know much about what I've written over the years, I'm prone to argue that heritability per se doesn't really mean very much, that it's omnipresent, it doesn't tell you how genetic something is, that the familiar terms that come out of behavior genetic models, A, C, and E, have a lot of other meanings than the traditional literal genes and environment. And I've even gone so far as to say there's no point in doing more simple heritability studies. And that often led to a question when I would say that, sort of, okay, smart guy, if we can't do our twin studies anymore, what should we do? Is behavior genetics over? And no, I don't think it is. And I'm a twin psychologist myself and a behavior geneticist. And so this talk is really about how can you use twins once you give up on heritability. And the core message is that most human psychology, at least of the kind I do, if you're not an experimental psychologist, is about trying to infer causation from non-experimental data. Developmental psychology in particular, and most of the studies I'll talk about are developmental, not all of them. Experimental control is usually impossible. So we observe correlations out in the world. We observe that some characteristic of parents is correlated with some characteristic in children. And what we wonder is that relationship causal? And so the way I see twin studies is as a tool to try and infer causation from correlation and non-experimental data. We talk about quasi-experimentation and quasi-causation, which are very old concepts in psychology, where psychology has a lot of roots in randomized experimentation. And in the 50s and 60s, scientific psychology was moving away from traditional lab-based brass instrument kind of psychology to more work out in the field. And it led to a lot of problems of how can we do valid causal inference on these kinds of data. And there were books written, Campbell and Stanley, that I was raised in grad school on about quasi-experimentation, which are imperfect but practical ways to infer something like causality from non-experimental data. I always like to start with this example. Not my own study, it's getting to be kind of an old study, but an example of the kind of correlation that you can observe in the world is the correlation between breastfeeding of children and various characteristics in the kids. And people always want to know, is breastfeeding good for children, or is not breastfeeding bad for children? And in particular, people have sometimes thought that breastfeeding might be related to cognitive development and even to adult intelligence. And this study was by a bunch of Danish researchers who found a data set that had recorded how many weeks of breastfeeding children had, and then their adult IQ. And So they set about looking to see whether there was a correlation between how long these people were breastfed and their eventual intelligence. And sure enough, there was. In the lowest group, there was a mean IQ of 98, and then at least in the 7 to 9 week group, a mean IQ of about 110, which is, a 10-point difference, which is no small thing. But of course, the problem is causality and knowing whether the breastfeeding is actually causing the increase in IQ, because children are not randomly assigned to breastfeeding conditions, and the mothers who choose to breastfeed are different in all sorts of ways from mothers who don't breastfeed. Among other things, they're richer and better educated. And so that could be confounding this relationship in a non-causal way. Well, it turns out that the National Longitudinal Study of Youth, a big population-based study in the US, has the same data. It has weeks of breastfeeding and IQ actually in kids, not when they're adults. And it has one additional piece of information. It has multiple children per mother. It has 3,000 children to 1,800 mothers. So there are siblings in the design. And mothers don't breastfeed all of their children the same length of time. So that allows us to do a very particular kind of analysis between mothers and within mothers. The between mother analysis is about averages. Do mothers who breastfeed longer on average have children who are more intelligent on average? Whereas the within mother is about deviations within families. Are the siblings who are breastfed longer more intelligent than the siblings who are breastfed less? And you get a very interesting result. Between parents, averages to averages, you replicate the Danish effect. 1.5 IQ points per 10 weeks of breastfeeding. Counts for 6% of the variance. Within mothers, on the other hand, there's nothing at all. There's no tendency for the kid who's breastfed longer to be smarter than the kid who's breastfed less. Now, what do we make of that? Well, I think it's intuitive that because children are not randomly assigned, it's a little bit deceptive to talk about what we're observing here as breastfeeding. It's not just breastfeeding. What we're observing is breastfeeding and between mothers, everything that's correlated with breastfeeding between mothers. So that is, for example, socioeconomic status and maternal education, and heaven knows what else. Hundreds of variables that we don't measure that differ among breastfeeding and non-breastfeeding mothers. Most of those confounding variables are controlled within mothers because it's the same mother. It's a fixed effect model. And so to the extent you see the effect between, but you don't see the effect within, what does that tell you? It tells you that the effect is coming from the confounds, it's not coming from the breastfeeding. We've spent the last 20 years in our twin lab identifying interesting correlations out in the world and then seeing how they hold up in twins. And a cool thing about doing this is that it turns out that most of developmental psychology and other kinds of psychology is waiting to be redone in a more causally informative way. Some very quick examples, because I don't have a ton of time. One interesting correlation out there is between step parenting and menarche, age of menarche in girls. It turns out that girls who are exposed to a stepfather tend to have earlier menarche than girls who are raised by their biological father. That's a well-known observation. It's a correlation. And it has as usual, girls are not randomly assigned to step-parent conditions. So there's a causal question. Why does that happen? Is it that step-parenting causes early menarche? There are hypotheses about why it happens. Early menarche is mostly correlated with negative things. I won't worry about that. In the adolescent health data set in the United States, they have information on step parenting and menarche in children. This is what's called a children of twins design. It's actually the mothers who are twins, not the children. So you can compare identical twin mothers, one of whom had a stepfather in the family and the other one did not. One mother remarried to a stepfather, the other didn't, and compare the daughters in those families. Now, between families, you, as before, you see the effect. That is, overall, girls raised by stepfathers had 6 months earlier menarche than girls not raised by stepfathers. But when you look inside the discordant families, that is, families where one mother had the stepfather, the other mother did not have the stepfather, there's no difference in menarche between their daughters. So once again, that shows that the effect happens between families, not within, which suggests it's a matter of some kind of confound, and later parts of the investigation are about identifying that confound, rather than anything specifically and causal about the stepfathering. Another interesting one, also from Adolescent Health, that I did with Paige Harden. This was a study that was published by a couple of sociologists about the Adolescent Health Project, showing that girls who in wave 2 of Ad Health had initiated sexual activity younger were more likely at wave 3 to be engaging in delinquent behavior. These sociologists gave it a hard causal interpretation and said, well, we should have programs to prevent kids from initiating sex too early so that they're not at risk for delinquency. And same story. When you look at it within twin and sibling pairs, the effect completely goes away. This is between where the girls on the right who started sex early are more delinquent. Within pairs, that is the sister who starts sex early more likely to be delinquent than the sister who starts sex late? Not only is that effect go away, there's a significant effect in the opposite direction. We don't really know what that means, but there's no evidence for a causal link between timing of first sex and delinquency. One more. This one, not a developmental study. One a little more recent that I did with some colleagues at the Washington State Twin Register, a big community-based sample of twins in the state of Washington, about an old nutrition problem, epidemiological nutrition problem, about trying to show that soda consumption is related to BMI. And sugary sodas obviously have a lot of empty calories in them. They ought to be related to BMI, one would think, but it's very, very hard to show. Why? Because people aren't randomly assigned to soda conditions, right? People who drink a lot of soda are different in a zillion ways from people who don't drink a lot of soda. That's just descriptive data. I haven't talked much about it. The identical twin correlation is higher than the fraternal twin correlation for both BMI and soda drinking, just like it always is. And this is the more numerical as opposed to graphical way we order the data. On the bottom line, labeled BP, these are the unstandardized regression coefficients relating BMI to how many soda portions you have a day. And it's a substantial and significant effect, about a unit of BMI per additional soda a day. But on the other hand, when you in the quasi-causal model, control for twin variables, which is essentially doing it within pairs, the effect is much attenuated, is no longer significant, and I mean, it doesn't quite go to zero, but it gets very, very small. So once again, what looks like a causal association out in the phenotypic world doesn't look so causal when you look at it within twin pairs. That was my time. Oh, it's getting bad. I think I'll skip the graphical part. That's the same result. What does all this mean? Behavior genetics isn't about estimating heritability. I just gave an entire, if fast, talk about BG without mentioning heritability at all and presenting my results. These models were capable of estimating heritability, but that's not the point of the study. The point of the study is to examine phenotypic relations while controlling for heritability, while controlling for genetic background. We always start with phenotypic associations. The basic tool of the field psychologist is observing correlations in the world and then examining those correlations for their causal status. BG models control for the environment as well as genes. That's sort of a nice thing about them. We think of behavior genetic models as this genetic tool, but it also controls for social class. If rich children have sex later and are less delinquent, that's a shared environmental confound of the causal relationship. So it's sort of nice to know that behavior genetics is a tool for studying the environment as well as for studying genes. Don't be literal about ACE. You know, when you have a significant A term in your twin study, it's not correct to say we've discovered something about the genetic components underlying X. So kind of get me off in a different direction. But ACE terms are domains of variance. They're not genes and environment. And more or less final message, behavior genetics is social science. Social science in general is trying to infer causation under very difficult conditions. And behavior genetics, twin studies, and really polygenic risk scores are exactly the same, is another tool in the quasi-experimental toolbox. It's no more a threat to psychology than the cross-leg panel design is. It makes causality harder to infer, but that's a good thing. We want causality to be hard to infer, and we expect that most of our correlations aren't going to turn out to be causal. I don't want to repeat myself. Behavior genetics is not heritability. I'll just leave that on the slide there. I refer to genetically informal designs as the humble revolution. Enthusiasts for behavior genetics often say, you know, genetically informative designs are a game changer because they're going to show us the right way to do psychology and that we have to take genetics into account. I think that's overstated. Behavior genetics is not about finding the genetic causes of things we used to think of as psychological. It's about using the structure of twin genetics to allow us to infer causality in certain ways that we can't infer it in our ordinary data. It's treated humbly. Behavior genetics is a way of doing old-fashioned psychology with just a little bit more attention to the subtleties of causation in our data. And that's a really good thing. And it's a way for behavior genetics to be part of mainstream psychology instead of in opposition to mainstream psychology. And I will stop my screen share and leave you with that thought. Thank you very much.
Thanks a lot, Professor. Fantastic talk. Very interesting, very groundbreaking. Very, very, very interesting. We are now open to questions. So if anyone has any question, you can put here on the chat on YouTube. We already has one first question for Professor Pluis from Fernanda Glaser. Have you found data related to changes in environmental sensitivity in the same individual, suggesting that he could be trained to change sensitivity according to his psychological needs? And I will add to this question if you think that would be possible to have some kind of training program for children in order to improve one thing or the other. I think it's related to that.
Well, it's a great question. We do not have much data on the continuity and the development of sensitivity, but we do expect that there will be some change. But yeah, at the moment, I don't have much to say about the level of change that we could expect. I do think my current understanding is that there is a genetic component to sensitivity, and if someone scores fairly low on their genetic sensitivity, then probably that individual will be less likely to acquire sensitivity over time. But for someone that is genetically prepared, they might have more scope to develop sensitivity. So we don't have any interventions yet that improve or reduce sensitivity. But the development of sensitivity is something that we hope to be able to look at soon.
Thank you, Professor. We have a question from Professor Otta to Professor Tukheimer. Thank you for accepting our invitation and for your excellent talk. I have a question for you. In 2015, a very complete meta-analysis was published of 50 years of research with twins at Nature Genetics. This meta-analysis is titled Meta-analysis of the Heritable of Human Traits based on 50 years of twin studies. Imagine that in 20, 30 years, a new meta-analysis will be published based on twin studies. You told us that heritability does not mean very much. Behavior genetics is not heritability. This new meta-analysis that will be published in 2050 will have a greater contribution of psychologists, sociologists, and researchers of humanities in general. Publications considered will be from research groups integrated by behavior geneticists and sociologists.
Well, I'm not exactly sure what to say about it. I mean, that meta-analysis was beautifully done. I recommend having a look at it. It's a lovely piece of work. I've always thought pretty much confirmed what I'd already said, which is that all human individual differences are heritable, and that's what the meta-analysis shows. And if you, I don't mean to be critical of it, but if you think about the way that meta-analysis has been used since it was conducted, it's cited as support for the idea that human differences are heritable in general, but the traits that happen to come out a little more heritable or a little less heritable in that study haven't really led anywhere. I mean, I think the highest the type of outcome with the highest heritability were ophthalmological characteristics. And what does that mean? I mean, does that mean that eye doctors should be rushing to the gene labs in ways that other scientists shouldn't? I don't really think so. That the things that make heritabilities high and low don't really have much to do with how important genes are for those traits. It's always important to remember, it's a standard example, that the heritability of how many arms you have is 0. And the reason for that is not that genes don't have anything to do with how many arms you have. The reason for that is that the genes relevant to how many arms you have don't vary very much among people, whereas the things that do vary among people to cause differences in arm number are mostly accidents and therefore environmental. And what that reminds us of is that what people for 100 years have wanted heritability coefficients to do, which is to be a measure of how genetic something is. They're not that. They don't work for that. Before we started to talk, there was a discussion on where heritability coefficients come from in the previous session, and I'm writing a paper about that. And just by the way, it goes back to Ronald Fisher, and then really invented the concept and was named by an animal breeder named J. Lush. But anyway, that's my thoughts on that meta-analysis.
Thank you. I have a question for Professor Pluis. I've been working with environmental sensitivity now for 2 years now. I'm trying to translate we talked, you probably don't remember, but we exchanged some emails. I'm trying to adapt your questionnaire to Brazil. And one thing that I kept thinking is you talk a lot about how the environment is perceived. And do you know any study with people with cognitive disabilities, or I don't know, people in the autism spectrum that are very sensitive to the environmental, to the environment, and at the same time they are not they don't have a lot of behavioral plasticity? And do you know anything about it?
I don't have specific data on that, but obviously, so we differentiate between, so sensory sensitivity can be observed in many cases in autism. People have heightened sensory sensitivity. What differentiates sensitivity as environmental sensitivity from that is that it's not just being sensitive to the environment by picking up on the sensory stimulation, but also the processing of that. So I think the current measure may not distinguish sufficiently well from that, and I think it's important to do more work on trying to figure out what drives the sensitivity in an individual, and is it just sensory sensitivity, or is there also that deeper processing of that? So what we see in sensitive people is that they pick up more what happens around them, but they also keep thinking about it for longer and process it. And that's something you have to as well see. And also the feeling overwhelmed by sensory stimulation would depend still very much on the context for sensitive people. So there's certain situations, maybe social situations that are there's still a lot going on, but it might be more manageable by a sensitive person, which someone with autism might still find it very difficult. But yeah, more work needs to be done to differentiate the sensory sensitivity component that we see as a normal common trait of sensitivity from what we see in autism. And a colleague of mine has run some analysis in TED study to look at the overlap, and there is some overlap, but I think the study design is correlational, so therefore it doesn't tell us that much.
Thank you. We have a question from Mateus Lima to Professor Tuckheimer. The research with genomes and the brain is still very expensive, but can you see those relations and variables being used to guide public policies in the long run with advances like Neuralink?
I don't know very much about Neuralink per se, so I don't want to comment on that. I do not see many domains outside of clinical medicine. I don't want to comment on heart disease, but in the behavioral domain, I do not see many real world applications of genetics, to the things that are often talked about, say education. I think on any way you frame applying, probably wouldn't be twins, polygenic scores to educational applications. I think, the relationships are fairly weak and not very likely to be effective, and B, to the extent they are effective, I think they're very likely to be discriminatory. So, though I'd be willing to consider any particular example somebody has, I, in general, don't think trying to use genetic data rather than phenotypic data. If you think kids' intelligence test scores, say, are important to how they perform in school, give them an intelligence test. Don't use their polygenic score that's correlated 0.15 with their intelligence test. And everything we know nowadays about indirect effects that underlie those just make it all the more problematic. So I'm pretty skeptical of real world application of these kind of data.
Thank you. And again, Professor Pluis, you talk about environmental sensitivity in a positive and negative way, right? And we probably have some genetic influence, but also developmental influences. Do you think that's a question I have reading the literature about that. Do you think that positive and negative sensitivity really do go together, or maybe because of some developmental issues, something can be separated? In my sample, I'm saying that I separated. For one group, I did a Latin class analysis. I get the 3 groups, but for one group, for 2 groups, it was the low in both, high in both. But in the other group, they were high on negative and low in positive. Do you think that this can have something to do with developmental questions or something like that?
Yeah, I think there might be different types of sensitivity, and that might very much reflect the specific history that someone has. So it would make sense from an evolutionary perspective that if someone has a genetic predisposition to be highly sensitive, that their sensitivity would match to the specific conditions that they're living in. So if someone grows up in a more challenging environment where there's a lot of threat, it would make sense that the sensitivity is shaped towards the detection of threat rather than the detection of opportunities. And the same way, someone growing up in a very caring environment, they may develop their sensitivity more towards picking up on sort of positive aspects. So we do think that there is sort of a programming effect of the quality of the environment for people that have this genetic predisposition for sensitivity. But we don't have a lot of work on that yet. But there is some work to suggest that there are those differences. And certainly we do find different interaction patterns. We do find certain people that show this vulnerability, but they don't show the vantage sensitivity. We do find certain people show the vantage sensitivity, but not the vulnerability. So I think it's very likely that the specific developmental history that individuals had, but also the current context that they're finding themselves in, would have an impact on the nature of their sensitivity.
Thank you. There's some questions coming here. Again, for Professor Pluis, can you tell something about, there's from Julia first. You showed the link between sensitivity and amygdala. Do you know any studies on mirror neuron system and sensitivity? Mirror neuron system and sensitivity? If you know, if not, what do you think about it?
So I don't know any study that looked specifically at mirror neurons and sensitivity. What I'm aware of is that although the whole idea of mirror neurons is very popular, I think there's quite a bit of debate about what that is and how it functions. There is, I think, quite a few people have written outside of the academic world about sensitivity being about mirror neurons. There is not really any evidence at this stage that I'm aware of. But we do know that sensitive people have a higher degree of empathy. So it would make sense if mirror neurons exist in the way that we think they might exist, that sensitive people would show a stronger reaction in those as well. But at the moment, I think that's pure speculation, and we don't really have any evidence to suggest that that is the case. But if it's mirror neurons that drive empathy, then it's likely that would drive that as well, particularly in sensitive individuals who tend to have higher empathy.
Thank you. From Nara Cortes, Professor Tuckheimer, thanks for your wonderful speech. Could you comment about how behavior genetics and epigenetic studies would interact to clarify causal relationship in developmental psychology?
Well, I mean, I think that's the way I think I see epigenetic studies, in the same way that I see linear genetics, that I don't expect that epigenetics is going to turn out to be a deep underlying explanation of phenotypic characteristics that we used to think of as psychological. So why are some people depressed? Well, it turns out that that's a result of epigenetic processes, either in their parents or in infancy or something like that. But rather, in particular cases, and my understanding of the epigenetic literature is that it applies much more to genuinely, in humans anyway, in genuinely traumatic situations, studies of starvation and war, that when you see downstream consequences, or at least purported consequences of things like that, where generations that were exposed to famine 50 years ago show deficits of various kinds today, it's the usual problem. People are not randomly assigned to famine conditions, and it's very, very difficult to assess the causality of the relationship. Epigenetic technology can be used to clarify the nature of those causal relations, and I don't think that's the same thing as trying to show that certain kinds of outcomes are caused by epigenetics.
Thank you. For Professor Pluis, from Suemi Tokumaru. Professor Plus, can you tell something about intra-individual sensitivity in relation to different environmental stimuli?
Okay, I'm not quite sure I understand the question. Individual sensitivity. Basically, I don't know whether that might be helpful for the person to clarify. Are you talking about individual differences within someone, whether they're sometimes more or less sensitive or sensitive to different aspects of the environment?
Well, we can go to the next one. Swami, if you want to clarify your question, you can write it on YouTube. I will get back to that. Professor Frowling also asked Professor Pluis, thank you for your very interesting presentation. I would like you to comment how environmental sensitivity relates to executive functions in managing and solving social problems.
Okay, so I can speculate on that. I don't have much specific on that, but I would say when it comes to solving social problems, being highly sensitive or scoring high on sensitivity has a range of advantages, but maybe also disadvantages. So the disadvantage would be that a highly sensitive person would perceive much more the negative components of a problem and might easily feel overwhelmed by that or feel threatened by that. The positive side is that a sensitive person is more likely to understand the social context, is probably more likely to be empathetic, to understand the other person, and therefore probably has an advantage in finding a solution that works for everyone. We do find in whether we measure sensitivity with our scale, but also when we indicate sensitivity with infant temperament or other measures, that sensitive individuals generally seem to have or develop a high social competence or social skills when growing up in a supportive environment, and that they have higher empathy. So I think that they would have developed executive functions, ideally, that would help them with that. But I would think that executive functions like self-control in insensitive individuals would be strongly impacted by the developmental history that they have. But certainly, I would say that it is possible for highly sensitive people to develop strong executive functions and that they can be very functional, especially if they grew up in a supportive environment where they had the chance to acquire those skills.
Okay, I have a question for Professor Tuckheimer. Today you talked a lot about within variation, and you focused a lot on that and questions of the behavioral genetics to control and try to aim us to a path where we can infer causation. But in other works, you also talked about the influence of the context, especially in poor environments. On IQ, for example, I remember you reading something from you that you say that if you are rich, if you have a good wealth and everything, your IQ is most influenced by genetic factors. But if you are poor, if you're in a poor environment, the context starts to be more important. And how do you think you can manage to deal with these aspects in the models you brought to us? That was mostly within. Your microphone is closed.
There. Well, I mean, I don't think they're at odds at all. It's important to remember that in my G by E for SES work, for example, that though I originally did it in terms of AC and E, that's not, in fact, how I do it anymore. In our most recent studies, we do it in terms of twin correlations. So if the genetic component in a twin study goes up with increasing SES, if A is going up, that means either the identical twin correlation is going up or the fraternal twin correlation is going down, or both. And it actually turns out reasonably consistently across studies that what's going on is that fraternal twins are more different in favorable environments, that DZ twins become more different, and that's what makes heritability go up. And then if you think about what's a correlation coefficient? I mean, these are intra-class correlations, which are a ratio of the between pair variance and the within pair variance. So twin genetics in general, all the way back to Fisher, is about comparing processes between and within pairs. And our most recent work in the Louisville Twin Study shows that in high SES families, what happens is that developmental processes within DZ twin pairs makes them diverge over time, as we think, this is a hard model, but as we think that fraternal twins who have small genetic advantages to lead them to have better phenotypes get systematically exposed to better environments, which makes them more different from their fraternal twin, and slowly but surely that process causes them to diverge. Whereas in impoverished families, those relatively better environments don't exist. So it doesn't matter if you, for genetic or phenotypic reasons, have a slightly higher IQ, there's no better classroom to get put in. There are no books for you to go seek out on the family shelf, so those processes don't take place. So I think all twin genetics is about between and within pair processes. And add one thing quickly, so is modern GWAS. What has the most contemporary GWAS turned out to be about? It's about comparing the performance of polygenic scores across families in the population to how they perform within pairs of siblings. And of course, for most behavioral traits, they perform much worse within pairs of siblings than they do in the population. And that's just showing in a new way what we've known from twin studies for years, which is that the effects of genes, such as they don't operate exclusively through the biology of the individual. They also operate via pathways that run through the family and social class and all those other things.
Okay, thank you. Thank you. I'd like to thank again Professors Lewis and Turkheimer. Wonderful talk, wonderful session. Many thanks. I think now we will stop for lunch.
I only wish we could have been there in person.
Sorry?
I only wish we could have been there in person.
Oh, yeah, we too. We really do too. So next year, maybe.
Yes, okay, good.
Near future. Let's end this damn pandemic thing, and then we will always be together. We all be together. So thank you again. I don't know if you want to say so. I think you want to say something about lunch. Come back at 2.
Yes, we will continue this. So we finish this section, but we will continue until 6 p.m. So we'll have a short pause for lunch, as Vinicius told us, but we will be back at 2 o'clock. I hope you will all be here with us. And just to remember you that the link for the YouTube for this afternoon will be a different one. And I just wrote it here in the chat, YouTube chat. So I thank you very much, Professor Plus, Professor Turkheimer, for the wonderful presentation. It was very nice having you here with us. Thank you.
Thank you. Thanks
Thank
a
you.
lot.
See you at 2.
All the best.